and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper.
Welcome everyone to our FDA Clinical Investigator Training course, better known as CITC 2024. Good day, good afternoon, or good night, depending on where you may be. It is so great and gratifying to see so many of you online with us here today, and we certainly appreciate your commitment in attending this event from wherever you may be. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance Program, better known as SBIA, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my extreme pleasure, along with my FDA colleagues, to be here with you today. And since 2018, it's been SBIA's privilege to collaborate with CDER's Office of Medical Policy to host this event. We bring you an unsurpassed cast of subject matter experts discussing a variety of topics which cross centers and which were specifically selected and designed for this audience. We are also offering live YouTube streaming, so please go ahead and invite your colleagues. We are extremely pleased to announce that this 3-day conference in its totality offers 12 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the registration process. Please note the emphasis on real-time and registration. And what this means is that no healthcare professional CEs can be given to those who attend without registration or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers, and the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete a final activity evaluation via the CE portal, and that's ceportal.fda.gov. And for multi-day activities such as this, participants must attest to their attendance and complete a faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity. No exceptions. So in this case, the deadline is December 26th. We apologize in advance, but we are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 or 7-digit profile number assigned by the CPE monitor, and your date birth should be in the month, day format. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print their statement of credit. Pharmacists should log into the CPE monitor 6 weeks after the last session of the activity to obtain their CE credit. Please allow sufficient time between entering the data before contacting us. To simplify things, each night's SBIA will send one email to the attendees of that individual event day, and that email will have information on both the CE claim code and the survey link. I strongly encourage you to complete the evaluation or evaluations upon receiving that email, especially in this holiday season with everyone taking vacation, as we will not be able to assist you if you miss the deadline of December 26th. Here are our learning objectives, and I'll give you a few seconds to peruse that on your own. As of this morning, we actually have over 8,000 registrants representing 120 countries. Thank you so much. 48% of these registrants have self-identified as small businesses, and of those, over 30% report having 1 to 9 employees. On this slide, we have our QR codes for our social media avenues, and the slide will also be displayed during breaks where you can scan them with your phone. Now let's turn our attention to some logistics. Here is the link for a one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate attendance, and so much more. It's the same page that you used this morning to access this event, so please bookmark it as you'll need it over the next 3 days. On our one-stop page, there are session evaluation links and also our final evaluation/survey, which will be activated this afternoon and will be sent in the daily post-event thank you email. Upon completion of the survey, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. The trick here is that the certificate must be downloaded immediately after completing the survey, otherwise that opportunity is lost. Again, the evaluation and certificate are only available for 2 weeks. So that means the deadline date for this is also December 26th. We made it easy for you guys, one date to remember. As a reminder, all the sessions are being recorded, and it will take us a good 2 weeks to process the recordings. But you will soon be able to access them from SBIA's homepage, and I sincerely hope that you have bookmarked that page, which is www.fda.gov/CEDAR-SBIA. In the interim, the YouTube link for each day will be posted on the One Stop Shop webpage at the end of that day. Just a few seconds here to address our upcoming SBIA events. January 22nd, we'll have a BSUFA 3 Regulatory Science Pilot Program and its progress update. January 28th and 29th, we'll have a knowledge management and modernization of regulatory quality assessment and submissions at FDA. And just a note that this is the first of its kind that we have that we're going to present in collaboration with CDER's Office of Pharmaceutical Quality. So please attend if that applies to you. And then April 9th and 10th, we'll have our Generic Drugs Forum, our annual Generic Drugs Forum. And this is an in-person event. And if you're interested, the hotel room block closes at on March 11th, 2025. So please go to our website if you need more information. Here is our promotion, which will be played during the breaks, so you can learn a little bit more about SBIA and what we do. And now I would like to draw your attention to this poll, which will be available during the breaks over the next 3 days. We would like to measure your interest in possibly attending this course in person. So please take a minute to complete the poll when it's displayed. We really do need your input. At this point, I would like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit. Special thanks to Drs. Leonard Sachs, Millie Dugal, and Kimberly Smith for designing the agenda and recruiting all our fabulous faculty. And they will also be our moderators throughout the event. Thanks to our speakers for their dedication and time, to our CE coordinators for working within such a tight deadline, and most of all, to you, the audience. And now I'm sure you're ready for us to launch fully into the day's agenda. I am extremely honored to introduce our host for today, Dr. Leonard Sachs. Dr. Sachs is the founder of CITC and has been the mastermind and driving force behind it. He joined FDA in 1998 as a medical reviewer in the Office of New Drugs. Subsequent positions included acting director of the Office of Critical Path Programs and associate director for clinical methodology in the Office of Medical Policy in the Center for Drug Evaluation and Research. In this capacity, he has led efforts to support novel approaches to clinical trials, including the use of electronic technology. Besides his involvement in the design and analysis of clinical trials, he maintains a special interest in tuberculosis and other tropical diseases and has published and presented extensively on these topics. Please help me to welcome Dr. Leonard Sachs.
Thank you very much, Brenda, for the kind introduction. Hello to everybody. I'm Leonard Sachs from the Office of Medical Policy in the Center for Drug Evaluation and Research at FDA, and I'm delighted to welcome you to this year's Clinical Investigator training course. As you all know, clinical trials are the bedrock for the approval of medical products, and clinical investigators are amongst our most important partners in developing the evidence used for regulatory decisions. We rely on you for all the clinical data that we receive to support the approval of new medical products, and we are tremendously appreciative of the critical contribution that you make to medical science and to the health of our society. This Clinical Investigator training course is an important opportunity for us at FDA to reach out to you with the goal of giving you a broad understanding of the science and the regulation of clinical trials. As many of you know, we have been running this course for years, and we are very encouraged by the tremendous support we've received. Today, as Brenda said, we have more than 8,000 registrants participating from all over the world, representing a global community dedicated to the development of the highest quality medical products. Our 3-day program covers a broad range of topics, including clinical trials, their design and analysis, the determination of drug safety, ethical considerations, and some of the innovative approaches to make trials more efficient and accessible to participants. We've included lectures on non-clinical drug development, animal studies, toxicology, chemistry, manufacturing controls, and we have an extensive session to address regulatory requirements, trial quality, and investigator responsibilities. We've divided the course into approximately 4-hour sessions spread over 3 days, and the idea is to cover the most topical areas of interest to investigators. Today, we will focus on selected topics related to the design of clinical trials, Tomorrow, we will look at some of the non-clinical and early clinical data that you may come across in the investigators brochure. And on Thursday, we will discuss the responsibilities of investigators and investigator site inspections. By sharing our experiences inside FDA, we're hoping to provide you with an insight into FDA requirements that will give you as investigators greater confidence and expertise in conducting clinical trials, and that will improve the clinical trial enterprise in general. I wanted to take this opportunity to thank our speakers in advance, all our colleagues of mine at FDA who have generously given their time to participate and prepare these presentations. I apologize for not being the most technologically gifted myself, but I hope we will be able to give you a satisfying experience as free of glitches as possible. During the panel sessions, we hope you will send in your questions to make this as interactive as we can. So without further ado, I'd like to introduce the speakers in our first session. Kim Smith will begin with an overview of FDA, its structure and mandate. Jim Smith will cover the basics of clinical trial design, and Mark Levinson will review the statistical principles for drug development. So Kim, over to you.
Thank you for that introduction. For the next 20 minutes, I'm going to provide you with an overview of the FDA structure and mandate to provide some background for later topics that we'll cover during the course. So the objectives of my talk are to provide a brief history of the FDA, to describe the current mission of FDA, and to introduce FDA's legal and regulatory framework for drugs and biologics. So to start with a brief history of the FDA, I think it's important to recognize that the agency was built on a legacy of public health failures, so unsafe, ineffective, counterfeit, or adulterated drugs, as well as some unethical practices and fraud. The image on the right is an advertisement for Mrs. Winslow's soothing syrup, which was marketed in North America and the United Kingdom in the 19th and early 20th centuries as a cure-all medicine for fussy babies. But the primary ingredients were morphine and alcohol, and thousands of children are believed to have died from overdoses or from morphine addiction and withdrawal. The lower right is an image from the New York Times. This is on the Tuskegee syphilis study, which was started in 1932 by the U.S. Public Health Service working with the Tuskegee Institute. It was a study to record the natural history of syphilis. Participants were not consented and were told that they were being treated for bad blood. By 1943, penicillin was the treatment of choice for syphilis and was widely available, but the participants were never told about this or offered treatment. In 1972, the study was exposed when an article was published by the Associated Press, but unfortunately at that time, men in the study had been untreated for many years and their disease had progressed. To continue with our brief history, I'll provide an overview of just some key milestones in U.S. food and drug law. So starting with 1848, we had the passage of the Drug Importation Act, which required U.S. Customs Services to inspect entry ports for the entry of adulterated drugs from overseas. It's really one of the earliest attempts to regulate the U.S. drug supply. Moving forward to the early 1900s, at that time, diphtheria patients were routinely treated with an antitoxin that was derived from the serum of horses. The horse serum was manufactured locally, and there were no central or uniform controls. In 1901, in St. Louis, Missouri, the blood of a tetanus-infected horse was used to develop the antitoxin and was administered to 13 children who ended up dying of tetanus. This tragedy led to the 1902 passage of the Biologics Control Act, which allowed for the first controls over processes used to produce biological products, including vaccines, serum, and antitoxins. In 1906, we had passage of the Pure Food and Drug Act, which prohibited interstate commerce in misbranded and adulterated foods, drinks, and drugs. In 1912, we had passage of the Shirley Amendment. The Shirley Amendment prohibited labeling medicines with false therapeutic claims intended to defraud the purchaser, but it was a standard that was very difficult to prove. Moving forward to the late 1930s, in 1937, we had another tragedy. At the time, sulfanilamide was a drug that was used to treat streptococcal infections, and there was demand to provide it in a liquid form. So a company experimented and found that sulfanilamide dissolved in diethylene glycol, compounded the product and sent it all over the country. Unfortunately, diethylene glycol is antifreeze and is toxic. The product killed 107 people, many of whom were children, and really provided a dramatic example of the need to establish safety before marketing a product. This tragedy led to the passage of the 1938 Food, Drug, and Cosmetic Act. The act required that new drugs be shown to be safe before marketing and really started a whole new system of drug regulation, and it was the origins of much of what we do today. In 1944, we had the Public Health Service Act. The Public Health Service Act covered a broad spectrum of health concerns and included provisions related to the regulation of biological products. In 1951, we had the Durham-Humphrey Amendments. The Durham-Humphrey Amendments defined the kinds of drugs that could not be used safely without medical supervision and restricted their sale to prescriptions by licensed practitioners. So this was our first foray into prescription products. We then have our next tragedy. Thalidomide in the 1950s and early 1960s was widely used to treat nausea in pregnant women. It became apparent in the 1960s that thalidomide was resulting in severe birth defects in thousands of children, limb deficiencies that you can see in the images on the screen. This was primarily occurring in Western Europe, and fortunately, the product had never been approved in the United States. Dr. Francis Kelsey, who was a FDA medical officer, had concerns about the amount of safety data that was available and had kept the drug off of the U.S. market. This example provided a lot of public support for stronger drug regulation and led to the 1962 passage of the Kefauver-Harris drug amendments. These amendments ensured drug efficacy and also enhanced safety controls. For the first time, drug manufacturers were required to prove to FDA that their product was effective before the product was marketed. In 1968, we had the Drug Efficacy Study Implementation, or DESI. Basically, many products had been approved between 1938 and 1962 on the basis of safety alone, and the agency had to generate a process to go back and review 4,000 plus drugs to determine whether they should stay on the market. The products that did stay on the market went through this DESI approval process. So fast forwarding a bit to the FDA now, the agency has over 18,000 employees, many of whom are scientists. Recent numbers are that over 11,000 of these 18,000 employees are in science fields. The agency oversees the safety of more than $3.6 trillion worth of food, tobacco, and medical products, accounting for 21 cents of every dollar spent in the U.S. On the right, you see a figure that was from 2023, although remains largely current. It shows that the human drugs program is the largest by far, followed by foods, tobacco, devices and radiological health, and then biologics. This course will really focus in on the human drugs and biologics programs. So I'm going to shift focus a bit to provide some information about the legal framework that guides the work that we do at FDA. Our work is grounded in statute with the Federal Food, Drug, and Cosmetic Act and Public Health Service Act providing our statutory authority for oversight of clinical investigations to evaluate safety and effectiveness. The laws are further explained through regulations that the agency issues. They can be found in the Code of Federal Regulations. This is how we implement the statutory authority over the conduct of clinical investigations. A few key sections. Title 21 is Food and Drugs, and Chapter 1 addresses the Food and Drug Administration. Subchapter A provides some general provisions. This is where you'll find information, for example, on informed consent and institutional review boards. Subchapter D addresses drugs for human use, and Subchapter F addresses biologics. FDA guidance is also issued regularly by the agency, and FDA guidance is intended to be advisory. It's intended to assist regulated entities in complying with the regulations and to help them to understand FDA's current thinking on a topic. There are numerous guidances, many of which are relevant to investigators, others might be more relevant to sponsors, and if you're interested in whether there's guidance on a particular topic, the link at the bottom provides the search location. So much of the agency's work revolves around review of submissions to certain types of applications. The first is the investigational new drug application, or IND, and then you have the new drug applications, NDA, or biologics license application, or BLA, and I'll talk a little bit more about each of these. So current federal law requires that a drug be the subject of an approved marketing application before it's distributed across state lines. Because the sponsor probably will want to ship the drug to clinical investigators in many states, it needs to seek an exemption from that legal requirement. And an IND is the means through which they technically obtain that exemption from the FDA, which allows them to ship the product across state lines. This, of course, allows them to initiate clinical studies in humans. There are three main types: investigator-initiated INDs, which I think would be the most common in most situations where you're studying a drug to meet an approval standard. There are also emergency use INDs as well as treatment INDs. There are two categories. There are commercial INDs. These are the ones that are initiated by a sponsor who intends to eventually seek some sort of a labeling change or approval of a product related to the studies that are conducted under that IND. For studies that are being conducted more for academic or research purposes, there are also research INDs. All INDs must include preclinical data, manufacturing information, as well as clinical protocols and information on investigators that will allow the agency to evaluate whether the study that's proposed is reasonably safe to proceed. Not all studies of drugs need to be conducted under IND. There are exemptions for specific types of studies. So first, the product has to be lawfully marketed in the United States already. This cannot be a new product that is not already on the market. The study cannot be intended to support a new indication or a significant labeling change for the drug. The study itself cannot involve a route of administration, a dose, a patient population, or some other factor that would significantly increase the risk to participants in the study. And the investigation needs to be conducted in compliance with other regulations for instance, institutional review board, informed consent, and promotion. So what is the purpose of an IND? The IND provides the data needed for FDA to ensure the safety and rights of clinical study participants are maintained, and also to ensure the quality and adequacy of studies intended to provide evidence of the safety and effectiveness of medical products. After a sponsor submits an IND opening study protocol, the agency has 30 days to review the protocol, at which point the protocol will go into effect unless the sponsor is notified earlier by FDA that the study may proceed or FDA issues a clinical hold. When the agency reviews these INDs or reviews any submission to an IND at any point in a development program, one lever we have if we have concerns about the study is to issue a clinical hold. This is an order that's issued by the FDA to delay a proposed clinical investigation or to suspend an investigation that is already ongoing. When we issue a clinical hold, no new subjects can be recruited or given the drug, and the patients in the study need to be taken off therapy unless they're permitted to continue by FDA in the interest of public patient safety. For instance, if stopping the drug abruptly would be harmful, there can be a plan to manage that in particular situations. Not all clinical holds are complete. There are partial clinical holds. For instance, if you have enough data to support lower doses but not higher doses, there may be a clinical hold on administering higher doses. Or for instance, if you have enough data to support studies in adults but not children, the studies could be allowed to proceed in adults while there's a clinical hold on studies in children. So what are the grounds for a clinical hold? For phase 1, 2, and 3 studies, the agency could find that there is really unreasonable and significant risk of illness or injury to participants in the study. There may not be enough information to understand the risk, so there may just be more information needed before a particular study or part of a study could move forward. The investigator brochure, which is intended to provide information to investigators on what is currently known about the drug, may be misleading, erroneous, or incomplete and need to be corrected to make sure that information on the drug is being accurately and completely portrayed to investigators who are enrolling patients in the study. The clinical investigators may not be qualified to conduct the investigation. And then lastly, if there is a disease that is life-threatening and affects both genders, but one gender, often women, are being excluded, that could be another ground for a clinical hold. For phase 2 and 3 studies, all of the above apply, but in this case, the agency can also put a study on clinical hold if the protocol is believed to be deficient in design to meet the objectives of the study. So moving on, once you have completed all of your studies under IND, you're ready for a marketing application. As I mentioned, there are 2 main types of marketing applications. One is a new drug application under the Food, Drug, and Cosmetic Act, and the other is a biologics license application under the Public Health Service Act. Both are a mechanism to provide the FDA with information that's needed to support the marketing approval of a new product, or to provide additional information to support a new indication or changes to labeling for an already approved product. Just to take a bit of a step back to a couple of basic definitions now that we're getting into new drug applications and biologics license applications. A drug is defined in the Food, Drug, and Cosmetic Act as articles recognized in the official United States Pharmacopoeia, official homeopathic pharmacopoeia of the United States, or the official national formulary. And they're articles that are intended to use in the diagnosis, cure, mitigation, treatment, or prevention of disease in man or other animals. And they're articles other than food intended to affect the structure or any function of the body of man or other animals. And then finally, they're articles intended for use as a component of something specified in A, B, or C. A biological product is a little bit different. It is defined in the Public Health Service Act as a virus, serum, toxin, antitoxin, vaccine, blood component, etc. Again, that's applicable to the prevention, treatment, or cure of a disease or condition in human beings. So all biological products would also meet the definition of a drug. So what do we receive in a marketing application, meaning an NDA or a BLA? First is chemistry, manufacturing, and controls information. So how is the product manufactured? How will it be packaged, et cetera? Non-clinical pharmacology and toxicology information, what is known about toxicities that were seen in preclinical studies or animal studies, human pharmacokinetics and bioavailability studies. So what is known about the clinical pharmacology characteristics of the product? Of note, later in this course, you'll have lectures on all 3 of these topics from some of the experts in these areas. For anti-infectives, there's information on the microbiology of the product. And then, of course, there's clinical information from studies in humans, clinical trials, and other types of studies, statistical information, as well as information related to potential pediatric use of the product. We get samples, as well as information on product labeling. And then for participants in trials, we do get case report forms describing events that may have happened during the studies, as well as data sets where we can actually reproduce some of the analyses necessary to confirm the efficacy and safety findings of the study, as well as to conduct additional analyses if needed. And then finally, we receive legal information. So information from the applicant about patents, exclusivity, financial certifications, and so on. Product. So the key decisions before us when we get either an NDA or a BLA are whether the drug is safe and effective in its proposed uses, and whether the benefit of the drugs outweigh the risks of the drug, whether the labeling is appropriate and what it should contain, so providers who read the label understand how to use the drug and who it would be beneficial in. And then finally, whether the manufacturing methods used to maintain the drug's quality are adequate and that the methods preserve the drug's identity, strength, quality, and purity. And so at the end of the day, if we find all of these to be true, we will approve a drug and it will be legally marketed at that point. I'll move on to the challenge question. So the first challenge question is for a drug that is lawfully marketed in the United States, an investigational new drug application is never required to conduct a clinical investigation using the drug. Is this true or false? So this is false. Not only does the drug have to be lawfully marketed in the U.S., but to be exempt from IND requirements, the study can't be intended to support a new indication or significant labeling change. It can't involve a route of administration, dose, patient population, et cetera, that would significantly increase risk. And the investigation needs to be conducted in compliance with other regulations, such as IRB and informed consent requirements. So challenge question number 2, which of the following is not a reason for a clinical hold? A, the investigator brochure is incomplete, B, there's insufficient information to assess risk, or C, the investigation of a drug for disease that manifests in childhood excludes children, or D, clinical investigators are not qualified, which is not a reason for a clinical hold. So C is not a reason for a clinical hold. So just to briefly summarize my talk, FDA has been working to promote and protect public health for over 100 years. It's one of our oldest consumer protection agencies. The laws, regulations, and guidance provide a framework for the development and approval of drug and biologics and really frames the work that we do day to day at FDA. INDs are the mechanism through which FDA evaluates whether clinical investigations are reasonably safe to proceed, and NDAs and BLAs are the mechanism for our oversight of the U.S. sale and marketing of drugs and biologics. Thank you very much for your attention. I will be back a bit later during the question and answer period, but now I will turn it over to Dr. Jim Smith.
Thank you very much, Dr. Smith. So as Kim said, my name is Jim Smith. I am the Director of the Office of New Drug Policy and the Office of New Drugs at FDA. And during this session, I'll be speaking for the next 45 minutes about the basics of clinical trial design. The learning objectives are to describe the characteristics of adequate and well-controlled studies, to describe the purpose of control groups and various types of controls, and to describe methods to reduce bias in clinical investigations. So to begin, and this might seem really basic, but we should keep in mind that the purpose of doing clinical research is to ask important questions and to answer them with appropriate studies. So the quote on this slide is the first sentence of the regulation that describes adequate and well-controlled studies, which are the characteristics of studies that should be submitted to FDA to demonstrate effectiveness for a drug. And it leads right off with a purpose. The purpose of conducting clinical investigations of a drug is to distinguish the effect of a drug from other influences, such as spontaneous change in the course of the disease, placebo effect, or biased observation. And although this regulation was specifically written to address, investigations seeking to demonstrate effectiveness of a drug, like I mentioned, the same principle applies to any study, frankly. One is generally interested in discerning the effect of some independent variable itself, like here the drug, on one or more dependent variables. That is the outcomes of interest. And if something other than the intervention being studied could have plausibly caused the outcome, then the study will have failed to answer the scientific question. So here I'm going to summarize at a high level the characteristics of an adequate and well-controlled study, which, as I just said, are good scientific principles to consider regardless of the type of clinical research that you're involved in. And I'm going to go into several of these points in the coming slides, but let me run through the list as an overview. First, studies should have clear objectives. So these should reflect the research questions, they should be clear and explicitly stated in a protocol, along with a summary of the methods that will be used to analyze the data to answer the questions. The study should use a design that permits a valid comparison with a control group, and that provides a quantitative assessment of the effect of the intervention. So I'll talk about examples of control groups shortly, as well as some common designs that are used to compare two or more groups in various ways. The selection of trial participants should be done in a manner that provides adequate assurance that they either have the disease or condition that's being studied, or in the case of a drug or intervention intended to prevent or reduce the risk for some outcome, the participants should at least be susceptible to having that outcome. Otherwise, there'd be really no reason for them to be subjected to the drug being studied. The method of assigning patients to the treatment and control group should minimize bias. This is where randomization usually comes into play for studies with a concurrent control, which I'll discuss in a bit. And beyond the method of treatment assignment, adequate measures should also be taken to minimize biases on subjects, observations, and analysts. Blinding is an example of such a measure. Next, there should be well-defined and reliable assessments of how the individuals in a trial may respond to an intervention. That is, what outcome or outcomes are being measured? Are we certain that we're measuring what we intend to be measuring? And last, there should be analyses of the results of the study that are adequate to assess the effects of the drug. These should generally be pre-specified in the protocol or statistical analysis plan before the study begins. So this may seem obvious, but it really can't be overstated. The scientific questions to be answered and the trial objectives need to align. So without having a really clear, specific question or set of questions, the study design choices are likely to be suboptimal, and then its results might be insufficient to answer the questions that you're trying to answer. So the questions that are driving the research should really inform choices about trial design, data collection, and analysis. So for example, if a new drug is being compared to a drug already on the market, is your question whether the new drug is clearly more effective than that existing therapy? Or would you also be interested in knowing whether it's at least not materially worse than the existing therapy, even if it's not clearly better? So each of those questions would not only require distinct analysis plans, but it would also impact sample size and possibly the importance of blinding and endpoint selection and other considerations. Or as another example, is the objective to understand the effective treatment when it's added to a specific background treatment, or would it be sufficient to understand its effect when it's added on to standard of care, which may or may not include a certain background treatment? The choice of a control group is always a critical decision. Control groups allow discrimination of patient outcomes caused by the test treatment from outcomes caused by other factors. So if the course of a disease were always the same in a given patient population, or if it was completely predictable from patient characteristics, then the observed outcomes that occur in a single treated group of patients, that is like a single arm trial, it could simply be compared with the outcomes that would have occurred in the absence of that treatment. And that is possible in certain circumstances, but typically a concurrent control group is needed because it's not possible to predict outcomes with adequate accuracy or certainty. And the choice of which control to use is important because it will affect the inferences that can be drawn from the trial, potentially the ethical acceptability of the trial, the degree to which bias can be minimized, the type of participants that can be recruited and how quickly, the types of endpoints that could be studied, the credibility of the results, and so on. So here are the 5 types of controls that are generally recognized in adequate and well-controlled investigations and are mentioned in the regulation that I cited. The first 4 are usually concurrent controls. That is, the control group and the test group are chosen from the same population and treated as part of the same trial. So the first, which I'm sure you've heard of, is the placebo control. So in a placebo-controlled trial, participants are randomly assigned to the test treatment or to an identical appearing treatment that does not contain the test drug. And it's very important to recognize, and sometimes this seems to be misunderstood, that the use of a placebo does not imply that the control group is untreated. In many placebo-controlled trials, the new drug and the placebo are each added to a common background therapy or other standard of care regimen. These trials are almost always double-blind, meaning that the patients and the investigators don't know what the treatment assignment is. And combined with the use of randomization and blinding, which I'll discuss a little bit more in a bit, this design actually controls for many, if not all, potential influences on the outcome other than those arising from the drug itself. So, for example, many diseases wax or wane in severity. So if a participant improves significantly after randomization, was that destined to happen regardless of the intervention, or, just because of the natural course of the disease, or was that a drug effect? Having a placebo arm can give you information about that. Or you might have heard about regression to the mean, where patients are eligible for a trial because they have, let's say, high blood pressure. So a high blood pressure reading gets them into the trial, but that might have been kind of a random high for them just from day-to-day variability. And in those cases, over time, even if without treatment, their blood pressure would usually fall back to that patient's average. And so having a placebo group can help assess for that and can show, what would have happened to a group in the absence of treatment. The second example is the no treatment control, and this is exactly what it sounds like. Participants would be randomly assigned to test treatment or nothing at all. That is not even a placebo. So really the main use of this design is when it's difficult or impossible to blind the treatment assignment. In the third example, participants are generally randomly assigned to different doses of the same drug or different regimens of the same drug. If a higher dose of the drug is shown to be more effective than a lower dose, that can provide really strong evidence that that higher dose is effective. The limitation, of course, is that the result would tell you something about that high dose, but if you haven't also included a placebo arm, you won't know whether or not the low dose is effective. Or similarly, if you don't see a difference between the high dose and low dose, then you're left wondering, are they both effective or are they both ineffective? For these reasons, it can be beneficial to include a placebo arm, which then provides information about a dose of 0, if you will. Next, in an active control trial, participants are generally randomly assigned to either the test treatment or a known effective treatment, which is referred to as the active control or the positive control. These trials can be double-blind by having participants in each arm receive not only their assigned treatment, but also a placebo version of the other arm's treatment, and that's referred to as a double dummy technique. So blinding in these trials is not always possible, though, because of some combination of either the number of interventions or different regimens or different routes of administration and so on. Last, in an externally controlled trial, a group of participants who received a test treatment are compared with a group that is external to the study. So maybe it was a group of patients observed at some time in the past, which is then referred to as a historical control, or it's a group observed around the same time, but in a different setting entirely, like a disease registry or a natural history study, or information collected from medical practice, and so on. Now, on occasion, an effect observed in a single-arm trial is known without question to be something that would not have occurred spontaneously. And so although this is sometimes called a baseline controlled study because a value on treatment is compared with the individual participant's baseline value, that's actually also a type of external control, because what you're really doing there is you're relying on the very strong knowledge, which is derived external to the trial itself, that the change that's been observed would not have occurred in the absence of the intervention. And there is a lot of interest right now in externally controlled trials, but it's really beyond the scope of my presentation today to cover them in sufficient detail. Suffice it to say that the lack of an internal concurrent control arm and randomization and blinding require exceptionally careful consideration of various ways that bias could be introduced into the trial and whether it's even possible to sufficiently mitigate these limitations to result in an interpretable trial. Okay, so moving on to the study population, that is, which patients are going to be included in the trial. So typically, the eligibility criteria or the inclusion and exclusion criteria should identify patients with or at risk for the disease or condition being studied. And this population might be more narrowly defined or more broadly defined depending on the objectives, which could vary during drug development. So early stage drug development, perhaps the population is being more narrowly defined because you want a more homogeneous population to detect a signal of a drug effect, or perhaps there's too much uncertainty about the safety of a drug in the early stages of drug development, that you need more exclusion criteria. But as that drug progresses to later stages of drug development, it becomes increasingly important to define the study population more broadly to more closely represent the diverse populations for which the drug is intended. And in general, you should avoid unnecessary exclusion criteria. So both to facilitate enrollment as well as generalizability of results, like each exclusion criterion should be very well considered and believed essential. And you should think about even commonly used exclusion criteria if they're actually necessary. Why exclude patients with renal impairment if the pharmacokinetics of the drug have been determined for those patients? Maybe they could be included, but with a drug with a dose adjustment, for example, or an age-related exclusion should be very well justified. So overall, the population that's expected to benefit from answering the research questions of interest ought to be represented in the clinical investigation, and enrollment goals should be proactively set to achieve that. So related to the study population, I'd like to briefly touch on the concept of enrichment. So this is the selection of a study population in which detection of drug effect, if one is present, is more likely than it would be in an unselected population. So this is a design feature that narrows the study population in certain ways in hopes of improving the ability to detect the effect of a drug. And so I'll go through three broad categories of enrichment strategies. So first, our strategies to decrease variability, because reducing variability often makes it easier to detect a signal among the reduced noise, if you will. So one could aim to reduce between patient variability or within patient variability. And an example of the latter is what I've included here. So a placebo lead-in period could be used in which all patients are provided a placebo for some amount of time before randomization. And those who show a large response to placebo, or those who demonstrate that they're very poorly adherent to taking the assigned placebo, could be excluded before they're ever randomized, as those participants might be might be more likely to introduce variability during the trial, making it more difficult to identify a drug effect. Another enrichment strategy is prognostic enrichment. So the question here is, who's most likely to progress or have an outcome of interest? So let's say you're conducting a trial in which the outcome of interest is hospitalization for heart failure, and the trial is designed to run until a certain number of such hospitalizations have occurred. Well, it could be quite beneficial to enroll patients who are at greater risk for hospitalization, like maybe those who have had a hospitalization for heart failure within 1 year of enrollment, or some other characteristic that informs their prognosis, which is why it's prognostic enrichment. The last strategy I'll mention is predictive enrichment. So here, the question is, who's more likely to respond to the drug than others without the factor that's being used for selection? So prognostic enrichment considers who's most likely to have an outcome regardless of treatment. And predictive enrichment considers who is predicted to respond to the drug. And so a randomized withdrawal study is an example of this. So here, all of the patients are placed on the drug for some period of time, and then a certain subset of those who believe they've responded to the drug by some predetermined criteria are then selected to be randomized to continue on the drug or to switch to a placebo. And that's an enrichment strategy because all of the patients who don't think that they're responders to the drug will have been weeded out, if you will. And they won't then be subjected to the test versus control randomized portion of the trial. Another example of predictive enrichment, maybe it's even more straightforward, is let's say you're enrolling patients who have a tumor tissue that's positive for a certain biomarker, such as a certain gene variant, and the drug's mechanism of action specifically targets that gene variant. That would be another example of predictive enrichment. Enrichment strategies can limit the generalizability of the findings, and I think Dr. Levinson might touch on this in his talk that's coming up. Okay, next we have method of assignment to treatment arms or study arms. This should be done in a manner that minimizes bias and ensures comparability of the groups with respect to pertinent variables. So typically, in a concurrently controlled study, assignment is by randomization. So why is randomization important? Let's think about a couple scenarios here. So in a randomized study, a patient who meets the eligibility criteria gets assigned to a group by a completely random method. So I'm illustrating that here by a flip of a coin, if you will. So heads, they get drug A, tails, they get drug B. So coins aren't actually used, but I think you get the point. So now think about an observational study where you go to the medical records of patients who are getting drug A and patients who are getting drug B, and you record things that you find in the record about their medical course, and then you compare the outcomes of interest of those who got drug A versus those who got drug B. So, well, why could that be a problem? Well, just highlighting one concern for illustration of this point, consider, how these patients each came to be on drug A versus drug B. So they may have come to their healthcare provider who took a whole host of things into consideration when making a treatment decision. Maybe the patient's age, their comorbidities, concomitant medications, family history, labs, the patient's own preferences, their insurance status, and so on. And if a difference observed between outcomes for drug A compared with drug B, if a difference is observed between those two, it's plausible that some other factor or factors other than the drug itself is associated with the selection of which drug to prescribe, but also with the outcome itself. So this could lead to an incorrect conclusion that there's a difference between drug A and drug B, when in fact, the difference is the result of the underlying patient populations. And this is referred to confounding, and I think that Dr. Levinson might be discussing this as well. So assuring that the subject populations are similar in the test and control groups is best attained by randomly dividing a single sample population into the test and control groups. So randomization is going to avoid systematic differences between groups with respect to both known and unknown baseline variables that could affect the outcome. And the balance of unknown baseline variables is an important one to keep in mind. It's not uncommon to see a great deal of hand-wringing over various baseline characteristics and their apparent balance or lack thereof in a randomized trial. And sometimes that is justified, especially when there's this known strong prognostic or predictive characteristic. But often it's not. In part, it should be kept in mind that there are a very, very large number of unmeasured baseline characteristics, and we have zero idea about their balance. Or lack thereof, because we have no idea what they, or their values are. Of course, this doesn't mean don't trust a randomized trial. In contrast, appreciate the ability of randomization to avoid these systematic differences between groups. And perhaps even more important, recognize that randomization provides a sound basis for statistical inference. And the reasons for that are beyond the scope of this presentation, but it really is a very important outcome or a reason for randomizing participants in a trial. So it's important that the control group be as similar as possible to the test group, except for the intervention that's being studied. To best assess the effect of the drug itself, the similarity between groups should be both at baseline as well as after baseline, through the study. And as I just discussed, randomization facilitates the similarity between groups at baseline. But many differences can arise after baseline, differences in adherence to assigned treatment, the use of rescue therapy perhaps, completeness of data and follow-up, and differences in how the outcome assessments are conducted, reported, and so on. And in the next few slides, I'll talk about methods to reduce bias entering the study during the trial itself. One of the key methods to reduce bias, at least in a randomized study, is to conceal the treatment assignments, which is referred to as blinding. So blinding is intended to minimize the impact of potential biases that could be conscious or unconscious. And as I just said, the aim here is to ensure that the study groups are treated and observed similarly during the trial, except for the intervention of interest. So what impacts could the knowledge of treatment assignment have? Well, I'll give several examples on this slide, but it's certainly not all-inclusive. And I'm also not saying that these will arise in every open label trial, but these are the types of things that should be considered and mitigated if possible. So healthcare providers could treat the trial participant differently if they know what study arm they've been assigned to. And those differences in how they're treated could directly affect the outcome of interest. So this would be another example of confounding. Those on active drug might report more favorable outcomes, such as responses on a questionnaire because they expect they should improve on the new drug being tested. Observers may be less likely to identify and report treatment responses in the control group, like the no treatment group. Those on active drug may be more or less likely to report adverse events. And this is a good reminder that oftentimes you don't know which direction these biases may go. Right? It's unpredictable. Knowledge may affect rigor of attempts to obtain follow-up data. And knowledge could impact treatment decisions related to analysis. Now, this isn't the investigator or the patients, but this could be a way that knowledge of treatment assignment could affect those other personnel, like those who are analyzing trial data. So that could impact their decisions, such as looking for reasons to justify excluding a poorly performing site. If getting rid of the results might make the rest of the trial results appear much more favorable for the test drug. And as I mentioned, there's no simple test for any of these. You can't really look after the fact and say, oh, well, this bias must have crept into the trial. And sometimes it'll favor the test drug. It's sometimes not, and you can't really assume that that's going to all balance out. So taking measures to reduce bias, especially in circumstances when blinding isn't possible, is really important. Now, even with blinding, it's important to realize that patients or investigators might be able to detect or guess their treatment assignment. Maybe there's a distinct adverse effect, that's only observed with the test drug, for example, or something similar to that. And in such cases, especially if the study is open label, one should consider pre-specified decision rules, blinded outcome assessments, and other measures. So as an example of a blinded outcome assessment, if the endpoint of an open label trial requires the histological assessment of tissue, or maybe the rating of a skin lesion from a photograph, the individual conducting the assessment should not be made aware of the treatment assignment. And this doesn't prevent all bias from entering the study, of course, but it would be an attempt to mitigate a biased assessment. Again, whether conscious or unconscious, by the study personnel who are aware of what treatment the participant is receiving. Knowledge of interim results, whether it's at the level of the individual or the treatment group, also has the potential to introduce bias or otherwise influence the conduct of the study and interpretation of its results. So special attention should be given to maintaining the confidentiality of interim results. And finalizing statistical analysis plans before treatment assignments are revealed is another mechanism to ensure that knowledge of the data itself does not influence the choice of analyses, or the choice of the inclusion or exclusion of certain data, the weight that's placed on certain endpoints, and so on. So shifting gears a bit, next I'd like to talk about assessing response in a clinical investigation, which is often described as the endpoints of the study. So the choice of endpoints should be aligned with the study objectives. And if the study is being conducted to answer the question of whether a drug is effective for a certain disease or condition, those endpoints should be meaningful for the intended population, and may also take into account the views of patients. The primary endpoint should provide relevant evidence related to the primary objective of the study. So, and typically the sample size of a trial is calculated based on information related to this endpoint. And then secondary endpoints are usually either supportive measurements related to the primary objective. That is, they provide additional information that might give one greater confidence in the results observed on the primary endpoint, or they may be measurements related to secondary objectives of the study. Usually the primary endpoint and at least some secondary endpoints are included in an analysis plan that protects against making false conclusions that an intervention has an effect when in truth it does not. Exploratory endpoints are those that may further explore or support study findings, or they may explore and generate new hypotheses for future research. So exploratory endpoints would generally not be included in the analysis plan in a manner that protects against making false conclusions. And because many potential endpoints and analyses could be explored, there's the very real chance that a difference between groups could be observed on one or more of these endpoints solely by chance alone. But what is an endpoint exactly? So this is a definition that comes from the current version of the FDA NIH BEST resource. BEST stands for Biomarkers, Endpoints, and Other Tools. So an endpoint is a precisely defined variable intended to reflect an outcome of interest that is statistically analyzed to address a particular research question. So when thinking about an endpoint, obviously you need to identify the assessment itself. For example, is it a lab value, a patient-reported outcome like a questionnaire, a physiologic test like maybe a pulmonary function test, or a clinical event like a stroke. Next, the endpoint might incorporate other information that occurs during the trial. So for example, if the endpoint is defined in terms of a success or failure, perhaps participants who discontinue their assigned study treatment prematurely are automatically considered a treatment failure. It should consider the timing of the assessment. So for example, are you interested in a hemoglobin value after 6 months of treatment? Or maybe the assessment is one that could happen at any time, such as a participant having an event like a stroke, and really the time of interest is the duration from randomization until when that first stroke occurs. And finally, if relevant, consider how assessments within each participant are formulated or combined. So for example, sometimes a composite endpoint is used, which means that multiple distinct events can be combined, if you will, into a single endpoint. Or maybe several assessments are made during the window of time, like a daily diary over a 2-week period. And you might decide in advance that you will take the average of all 14 scores, or maybe it's the maximum or the minimum, to represent the endpoint assessment in a single value. So there are different types of endpoints, and I've already alluded to several, but a couple major categories include clinical outcomes and surrogate endpoints. A clinical outcome is an outcome that describes or reflects how an individual feels, functions, or survives. That is, it directly represents the clinical state of an individual. A surrogate endpoint, on the other hand, is an endpoint that is used as a substitute for how a patient feels, functions, or survives. So an effect on the surrogate endpoint is expected to predict an effect on the ultimate clinical outcome of interest. And the level of evidence that an effect on the surrogate predicts such a clinical benefit can vary, of course. So if the evidence is strong, at least in drug development, that's called a validated surrogate endpoint. Examples of this could include reductions in hemoglobin A1C, which have been clearly shown to predict benefit with respect to microvascular complications and diabetes, or reductions in blood pressure, which reduce the risk of certain adverse cardiovascular events, such as stroke, among patients with hypertension. I'd also like to mention the idea of intercurrent events. So intercurrent events are events that occur after treatment initiation that affect either the interpretation or the existence of the measurements associated with the clinical question of interest. So some examples of these types of events could be treatment discontinuation, and that can occur for a variety of reasons, like adverse events, perceived lack of efficacy, other things. Beginning a new therapy or switching to other treatments, and which could affect the outcomes of interest, or even something like death. And it's important to note that intercurrent events do not lead to a problem of missing data. So missing data is data that would be meaningful for the analysis, but were not collected, which is different than data that do not exist or data that are not considered meaningful because of an intercurrent event. So for example, although missing data strategies is beyond the scope of what I'm talking about in this session, imputation techniques are often used to impute values for those that should have been collected but weren't. So consider a participant who dies during the trial before they reach the time point at which the endpoint would be measured. Well, they won't have an endpoint value, but it would usually not make any sense at all to impute a value for someone who died, essentially pretending that they were alive and had an endpoint measured. So in this case, death is an intercurrent event. And if it's predicted to happen with some frequency in a certain study population in a trial, how to handle such an intercurrent event should be considered and incorporated into the design and analysis plan before the trial even begins. So you should also consider possible intercurrent events when defining the clinical question of interest. So here's an example. In a trial of a drug for type 2 diabetes, suppose the primary endpoint is the change in hemoglobin A1C from baseline to week 24. And suppose the trial allows for rescue medication if certain thresholds are met with respect to blood glucose values being too far out of control, for example. So here are two examples of possible treatment effects that may be of interest. What's the effect of the new drug if rescue medication is not available? Or what is the effect of the new drug regardless of whether rescue medication is used? Now, I don't have time to discuss how the effect of interest would result in different design considerations, but to suffice it to say that these are distinct questions, right? And that should be considered on the front end, not after the fact. Okay, so the elements that I've discussed can be put together in various ways, and I'll just touch on a few here. The most common study design for a randomized controlled trial is the parallel group design. So in this design, participants are randomly assigned to different groups that are then followed in parallel, if you will, until the endpoint. Another study design, which still employs randomization, is the crossover design. So here, participants are randomly assigned to an initial study treatment, so test versus control in my illustration here. And after some period of time, endpoints are assessed. And then there's usually a washout period where no treatment is given, allowing the effects of the first period to, possibly return to their baseline values. And then within each of the initially randomly assigned groups, each participant then receives the other study treatment for a second period, and the endpoints are assessed again. And this design can be more efficient than the parallel group study and therefore require fewer patients because it has the advantage of each patient serving as their own control, which removes the between patient variability from the comparison between treatments. But there are several things to consider that might make this a suboptimal design. So first, there's the possibility of a carryover effect, which is the effect of the treatment from the previous time period carrying over and influencing the response during the subsequent time period. And that washout period in the middle is an attempt to mitigate this, but it's still possible that the previously administered treatment may have altered the individual in some way so that they'll react differently to the treatment from that time onward, or maybe the washout period is just, an insufficient amount of time or whatnot. Or maybe the outcome is some sort of a functional test that requires some skill, so that participants are typically perform better during period 2 on the task because they were trained in a sense during period 1. So those are things to watch out for. Second, the design is generally used when the observation periods are relatively short, so that the endpoint needs to be one that responds and can be measured fairly quickly. Also, since ideally each individual would return to their baseline condition before each period, it's generally used in chronic diseases where the outcome is not dependent on the time of the assessment. So last, I'll describe a 2x2 factorial design. So I've drawn the study arms here as a table, but this would typically be a parallel group study. But each participant gets randomly assigned, so in this case, to either placebo, drug A alone, drug B alone, or the combination of drug A and drug B. And of course, this could be blinded using the double dummy technique that I mentioned earlier. And this allows you to essentially study two interventions using the infrastructure of one trial. So first, you can assess the effect of drug A by comparing the two groups in the left column, that is placebo and drug B, with the two groups that include drug A, that is drug A alone and drug A plus drug B. Similarly, you can assess the effect of drug B by comparing the top row, so placebo, which, placebo and drug A, so it does not include drug B, to the bottom row, which does include drug B. But the design also allows other analyses. So for example, you can look at whether drug A and drug B each contribute to effectiveness when they are used in combination. You can also check whether the effect of drug A is modified in the presence of drug B and vice versa. Factorial trials can certainly include more than two interventions, but it can quickly become infeasible since the number of cells in a full factorial trial will simply become too large. Also related to the comparison between groups, and I've mentioned this already, is the question of whether you're testing a superiority or a non-inferiority hypothesis. So in a trial of drug A versus a known active control drug B, the superiority hypothesis is testing whether drug A is better than drug B. And by better, this would usually mean more effective, although I suppose you could test a hypothesis related to superior safety as well. In contrast, the non-inferiority hypothesis would be testing whether drug A is not meaningfully worse than drug B. And the non-inferiority margin, or NI margin, quantifies what is meant by meaningfully worse. Now, if no difference is detected between drug A and drug B in a trial, then there's a question. Does that mean that both drugs are effective or that both drugs are ineffective? And that's obviously a very important difference. And so because there's no confirmation, within that trial itself that the active control drug B is effective, that's an assumption. And so it must be considered very carefully. So assay sensitivity is the ability of the trial to have detected a difference between treatments of a specified size. So said another way, assay sensitivity means that, had the study included a placebo, the active drug would have been shown to have the effect that was assumed. And this is not directly assessed within a non-inferiority trial. So you need to consider such things as, historically, have consistent findings been observed with the active control, especially when compared against placebo in those past trials? If there has been a consistent effect, then you have greater confidence in the active control in the new trial. Second, how similar is the new trial to those historical trials where the active control drug was tested against placebo? Is the patient population similar? Were there important differences in concomitant treatments that have become available? Are the differences in how the study endpoints are defined or analyzed? Is the active control being dosed similarly in the new trial as it was in the historical trial? So these are things to consider because you're making an assumption that the active control's effect is constant between the new trial and the historical trials, and that's referred to as the constancy assumption. Now, third, the quality of the new trial needs to be excellent. So poor conduct can reduce the ability to detect differences between groups because sloppy conduct generally introduces variability. So this can be problematic in a non-inferiority trial because the trial could be considered a success if the new drug and active control appear to behave similarly. And you won't know for sure whether that means that both drugs are effective or both drugs are ineffective if the trial was not conducted well. Finally, just a couple high-level comments about trial design and to some extent conduct. So efforts should really focus on activities that are essential to the study. So for example, it's far more important to expend energy and resources to ensure that the primary endpoint is assessed exactly as intended, that follow-up is as close as possible to 100%, rather than pouring resources into ensuring that every scrap of source data is available for the 50th exploratory endpoint. Obviously, I'm giving an extreme example there, but I'm just trying to emphasize that you should consider eliminating non-essential activities in data collection to increase quality by simplifying conduct, improving the efficiency of the study, and to really target resources to the critical areas. So deploy your resources to identify and prevent or control errors that matter. And last, clinical study design is best informed by input from a broad range of interested parties. So patients, patient organizations, study coordinators, site staff, clinical investigators, like these, people who will be directly involved in the successful completion of the study are very well positioned to inform its design. Patients may take one look at a protocol and say that it's far too burdensome in a variety of ways, and that could potentially foreshadow a lot of premature treatment discontinuation if it's not modified. Clinical investigators and site staff could give a lot of insight into the feasibility of enrolling patients who meet the inclusion and exclusion criteria, and maybe those need to be modified or broadened. Or whether the scheduled study visits and procedures are overly burdensome and might also lead to early dropouts. So and patients and patient organizations can inform the general relevance of study endpoints, study settings, and some among other things. So seek this information out and really consider using it to optimize the design of the study. So with that, I'd like to move on to my challenge questions. The first is, which of the following is not an example of enrichment? Enroll patients with a lab value that indicates they are at greater risk for the outcome of interest during the trial. Limit enrollment to patients with a certain genetic variant that is specifically targeted by the investigational drug. Ask trial participants who demonstrate poor adherence after randomization to withdraw from the trial. And require patients to have 3 consistent values during the screening period for the assessment of primary interest in the trial. Give you a few seconds to think about the answer. And C is not an example of enrichment. So A is an example of prognostic enrichment, B, predictive enrichment, D is a way to reduce variability, but C says to ask participants who demonstrate poor adherence after randomization to withdraw from the trial, and you do not want to do that because after randomization, pulling participants out with poor adherence could introduce bias. And so if you're going to use this technique, it should be done prior to randomization. The second challenge question, the choice of a control group affects which of the following? A, the inferences that can be drawn from the trial. B, the ethical acceptability of the trial. C, the degree to which bias in conducting and analyzing the trial can be minimized. D, the scientific credibility and impact of the results. And E, all of the above. And hopefully that's clear that that should be E, all of the above. So to summarize this session, you should design clinical investigations to distinguish the effects of an intervention from other influences. Always start with the clinical questions of interest, and the design of the clinical study should flow from those. Key design aspects include the study population, an appropriate control, well-defined endpoints, measures to minimize bias, including the assignment of treatment, and the analytical plan. And with that, I'd like to thank you very much for your attention, and I will turn it over to Dr. Levinson.
Good afternoon, everyone. My name is Mark Levinson. Today I'm going to be talking about statistical principles for clinical development. There are two learning objectives to my presentation. The first is to understand basic statistical concepts such as bias, variability, and multiplicity. And the second is to understand the role of statistical concepts in good study design, conduct, and analysis. So basically, the first learning objective will lead into the second. Here's my outline. First, I'm going to give a quick review of good study principles. Dr. Smith has already presented on much of this. And then the heart of my talk is I'm going to go into statistical concepts and the implications on study elements. And this will start with a discussion of the distinction between bias and variability and the implications of each. And then I'm going to drill down into variability and then bias. And finally, I'll provide some concluding statistical principles. Okay, just to get some terminology down that I'll be using throughout my presentations, I'd like to provide some terms for the stages of a study. The stages start at the design stage. This is the conception, planning, and specification of the study. Typically, this will result in the protocol and statistical analysis plan, or SAP. The second stage is the conduct. This is the running of the study. This includes recruitment, screening, intervention, concomitant care, outcome ascertainment, and safety and study monitoring. The next stage is the analysis of the study. This is the number crunching, going from the raw data to the final results. Typically, this is where people believe the statistician comes into play, but I argue that the statistician has a role in all the stages of a study. Finally, there's reporting. The reporting of the study should basically align with the protocol and statistical analysis plan. I won't go into reporting much today, but there are guidelines on this. A common theme or message in my presentation is that design and conduct are more important than analysis. In other words, analysis cannot make up for poor design and conduct. One should focus on good design and conduct, and the analysis will be straightforward. And statistical concepts motivate good design and conduct, and that's really the point of my talk, is to show where statistical concepts can improve design and conduct and result in overall a better study. So what are the goals of study design, conduct, and analysis? The first goal is very clear. You're trying to address a specific clinical question. That's why you're running the study. I'm going to present two additional goals that come into play as you choose design and conduct decisions. The first one is to minimize bias and minimize variability. I'm going to spend a lot of time on what that means, both for bias and variability. And finally, you have to be able to ethically, safely, and feasibly conduct the study. You can't do a study that's not ethical for patients, not safe for patients, or it's just not feasible. Now, the problem is there's some conflict among these goals. There's almost inevitably some conflict among these goals. And let me give you an example. Okay, here's an example of balancing these three goals. And this is the concept of enrichment versus generalizability. So the clinical question we're interested in is a drug effect on cardiovascular events for all patients with type 2 diabetes. And the emphasis here is on all patients. This is an event trial, so we're trying to measure cardiovascular events. Event trials usually require a certain number of events to get the necessary statistical precision. You can get more events for the same number of patients if you study patients with additional CV risk factors. For example, patients that may have had a prior heart attack. So for the same number of patients, if your patients are at greater risk, you're going to be able to run a smaller trial. However, you're not really answering the first clinical question that I stated because you're interested in all patients with type 2 diabetes. So here we see that there's a balance between feasibility and answering the clinical question. Okay, that was my quick review of good study principles. I'll return to many of these principles throughout the talk, and now I'm going to get into the heart of the presentation, and that is the statistical concepts and their implications on study elements. And first, I'm going to define bias and variability and the important distinction between the two. Okay, here we have four bullseyes. Let's start in the upper left, low variability and low bias. The dots are intended to represent your data points, and the center of the bullseye is intended to represent the truth, the answer you're trying to seek. So in this bullseye on the upper left, we see how data points are packed closely together. That's the low variability, and they're actually centered on the center of the bullseye, and that's the low bias. Moving to the upper right, we see that the data points are spread apart, and that's the high variability, but they're generally centered on the average around the center of the bullseye, and that's the low bias. Moving to the lower left, the data points here are clumped together with low variability, but they're far from the center of the bullseye, so this represents high bias. And finally, the combination of the two, in the lower right, we have the data points spread apart and not really centered around the middle of the bullseye, and that's high variability and high bias. So those basically informally define these two terms. Now the problem is you don't get to see the bullseyes. So say these were your data points, and I ask you the question, do these data have low bias? Well, I mean, let's say, are we able to even answer, do they have low variability? Well, that's a relative term, but using kind of the scale we had on the last slide, let's say these data points have low variability. But because we don't have the bullseye here, we don't know the true answer because that's why we're running the clinical study. We don't know if these data points are centered on the truth or centered around some value away from the truth. So we don't know about the bias in this case. I hope those few slides demonstrated a few things. First of all, I hope they demonstrated what bias and variability ultimately are, but there are some other important concepts I hope they demonstrated. First, that we see and can measure variability. We can see how the data points are spread apart from each other. We can measure that using such measures as standard deviation and ranges. We do not see bias. We don't know what the true answer is, or not, we wouldn't be running the study. Bias is mainly addressed in the design and conduct stages, and I hope the statistical principles I bring out through this talk will help motivate good design and conduct. Now we have a definition of bias and variability, and I'm going to be drilling down into each of them. First, variability and associated statistics and concepts related to that. So variability and sample size. So sample size, of course, is the number of people in the study. I'm going to ask you a question. What is a better estimate of the average age of this session's attendees? Now, there may be over 1,000 attendees in this class, and we would like the average of those 1,000 values, but we're going to take a sample of the attendees, as is often done. So the first possibility is we pick a random sample of 5 attendees and calculate their average age. The second choice is to pick a random sample of 50 attendees and calculate their average age. Now, which of this is going to give you a better average age estimate of the overall session attendees? Clearly B will, because more people are involved in this sample. Some people will have high values relative to the overall mean, some will have low, and the more you pick in your sample, the more there's going to be cancellation, and there's going to be less variability in these larger sample sizes. So Now I'm going to introduce a little notation to further explore variability in sample size. Let's call x hat 5 the mean of 5 people, and x hat 50 the mean of 50 people. We intuitively saw on the last slide that x hat 50 has less variability than x hat 5. Let's look at variability in sample size a little more mathematically. The standard error of x hat 50 is a measure of the variation of x hat 50. And the standard error is defined as the standard deviation of x. Those are the individual values of x, the individual ages in the population, divided by the square root of n, the sample size. So as you can see from this formula, as the sample size n gets larger, the denominator gets larger and the ratio gets smaller. So for larger sample sizes, the standard error decreases. Okay, we're going to get into a little bit more math. I'm going to introduce the z-statistic. The z-statistic is the effect estimate. So that's basically what you're trying to estimate, like the effect of a drug, over the standard error of the effect estimate. So this is an estimate over a standard error. It's basically a signal-to-noise quantity. Just to make this a little concrete, I'm going to assume the effect estimate is 0.5 meters. Now I'm going to be using kind of a running example through a few slides that the endpoint we're interested in is how far you can walk in 6 minutes. And what we've observed in this study is that there's a 0.5 meter greater distance that people on drug can walk than on control. That's a small amount, but that won't be important right now. As the sample size increases, we saw on the last slide that the standard error goes down. And as the standard error goes down, because it's the denominator in this quantity, the z-statistic goes up. And what I won't really show is as the z-statistic goes up, the p-value goes down. What that means is it's more significant. The same result, the 0.5 meters, if you observe that in a larger study, that will give you a more significant result. Let's continue this. Typically, if there is no drug effect, then the probability of seeing the absolute value of the z-statistic greater than 2 is 0.05. So 0.05 is 1 in 20. And absolute value means if we see -3 or +3, they're both greater than 2. If we observe an absolute value of the z-statistic greater than 2, then we say the finding was significant at the two-sided 0.05 level. So what this says is if there was no drug effect and we observed, say, a z-value of 3, that was a pretty unlikely event, certainly less than 1 in 20. And that gives us doubt that there really is no drug effect, and maybe the assumption of no drug effect is wrong. So we call that statistically significant. It's a tricky concept, but that's basically what it is. Now let's return to the effect of sample size. For a given real effect, say 0.5 meters, the probability of a significant effect increases with sample size. So we saw that on the last slide. If the effect is 0.5 from a larger study with more sample size, it will have a smaller p-value, more significant than the same effect from a small study. So when we say a larger study has more power than a smaller study, because the probability of seeing a statistical significant result for the same effect size is larger. So to say that again, larger sample sizes have higher power. Now I will move on to the concept of multiplicity, also known as multiple bites from the apple. Suppose I told you a study found a drug had a statistically significant effect for females aged 51 to 60 years old. Well, that sounds like an important finding. Now, suppose I told you the study actually studied people from all ages, from the first year of life to people over 90, and both males and females. And they calculated the z-statistic for every combination. So each of these cells gives you a chance of the z-statistic being greater than 2, even if there's no drug effect. So perhaps it's not surprising that some were greater than 2. In fact, greater than 2, even if there's no drug effect, occurs 1 in 20 in a 1 in 20 chance. So here it's not at all surprising that if we looked at enough of these different combinations, one of them would be greater than 2. But it doesn't, I think, call into question that there's no drug effect. It very likely could be no drug effect, and you would see a result like this. So multiplicity, even if a drug has no effect, if you conduct enough statistical tests without proper adjustment, you'll find some results statistically significant. This is known as multiplicity. And what the implication is here, that although they're nominally statistically significant, they're not really statistically significant. They're not unusual findings. So multiplicity can show up with multiple subgroups. So we saw age and sex a few slides ago, multiple endpoints or analyses. So subgroups might be effect on males, effect on females, effects on people over 65. Endpoints might be effect on blood pressure, effect on life expectancy, effect on happiness. And finally, if you even run the same different statistical analyses on the same subgroups and same endpoints, you may get different results. So for example, if your primary analysis is some sort of multiple regression, but use different covariates, you actually may get different results. So multiplicity can come up in a whole variety of ways. There are solutions to the multiplicity problem, and that the primary solution is pre-specification. You tell the world ahead of time what you will primarily look at. So in that study a few slides ago, if the study had primarily ahead of time decided that they're most interested in females of a certain age, and they found a statistically significant result in that subgroup, that would be an important finding. At least that would be a statistically significant finding. So this avoids what's known as fishing for p-values, looking at multiple subgroups or endpoints until you find something that has an interesting looking p-value. So the way you tell the world ahead of time is through protocols and statistical analysis plans. So those should be completed before the protocols should be completed before the start of the study, and the statistical analysis plan is usually completed before unblinding of any data. Now, there are statistical methods that can handle multiple outcomes or multiple subgroups. I mean, suppose you genuinely are interested in 2 different endpoints, like blood pressure and survival. There are statistical methods that can properly adjust just for these multiple comparisons and this multiplicity. But I certainly won't go into them today, but the important thing is they need to be pre-specified too. We spent a little time on variability, and now I'm going to move on to bias. So in bias, I'll cover various types of bias and say something about sensitivity analyses. This section will be longer than the variability section, but more intuitive, I hope. Okay, we'll go back to this formula for a z-statistic. There's nothing new here. We have a z-statistic that's the effect estimate over the standard error of the effect estimate. And for a given effect size, say 0.5 meters, if the sample size goes up, the standard error goes down, the z-statistics goes up, and the p-value goes down, meaning it's more significant. Let's look at a bias example. Here we have an unblinded trial, and the endpoint is effort dependent. I'm going to use the 6-minute walk example again, how far you can walk in 6 minutes. Because patients know what drug they're on, whether they're on the experimental drug or the control, patients on the experimental drug may try harder. this may not be a conscious bias. This may be more of an expectation bias. They think they're on some novel therapy, so they're going to be doing better. Let's say on the average that they do 0.5 meters better. Some may do more than 0.5 meters, some may do less, but on the average, this expectation bias makes people on the experimental drug do 0.5 meters longer in a 6-minute walk. Let's assume there's actually no drug effect here. Now, the important point here is this 0.5 meter bias will not likely be affected by sample size. Everybody will have, on the average, this 0.5 meter bias. It's going to appear no matter what sample size you have. So p-values will get smaller for larger sample sizes, even though the drug is not affected. We've learned something about addressing bias. Sample size does not address bias. A larger sample size will not get rid of bias in most cases. What you need is design and conduct. You use design and conduct to address bias. So some of the design methods are randomization, blinding, good outcome ascertainment. What I'd like to say is analysis cannot rescue poor design and conduct. Analysis can address some bias and explore sensitivities to bias, which I'll talk about at the end of the talk. So in order to help identify biases, it's useful to categorize them. So I'm going to categorize 3 types of biases. And it's not an exhaustive list, but it accounts for much of the bias you may see. The first is confounding. This has to do with baseline differences between treatment groups. And this is addressed by randomization. I'll go more into this. The second type of bias to have your eye out for is measurement bias, or the endpoint ascertainment. This is typically addressed by blinding and reliable and valid measures. I really won't be saying any more about this in today's talk. And finally, there's selection bias. This includes both baseline and post-baseline population issues, which I'll have a bunch to say about in a few slides. Confounding. So without randomization, there may be systematic differences, bias, when comparing people getting drug A and people getting drug B. This is known as confounding. Let's take an example of this. Suppose drug A may be given to older, sicker patients. Perhaps drug A has less side effects, and older and sicker patients can tolerate it better. So even if there was no difference between the effects of drug A and drug B, the comparisons may show that drug A has worse outcomes because drug A is made up of sicker patients. Again, this is known as confounding. Now, I already mentioned, randomization addresses confounding because it makes the 2 comparator groups comparable at baseline. Many of the studies you see in the paper are not based on randomized studies. So if you ever hear anything about coffee or red wine or chocolate, chances are it was not based on a randomized study, and I would take the results with a grain of salt. Now let's move on to selection bias, and I've broken that up into 2 types. So the first type is baseline study population is not representative of the intended population. But this is a relatively easy concept to understand. So let's take an example. So suppose in your study there are a few people with comorbidities. So this will affect the generalizability or the external validity of your study, that what you study may not represent the larger population, in this example, people with comorbidities. The second type of selection bias is that the analysis population is based on post-baseline information. This is harder to understand, and I'm going to spend some time on this. So some quick examples that we'll dive deeper into are analyzing adherers only, analyzing those with no missing data, and analyzing those with an initial response to treatment, you can see that the population you're analyzing is all based on post-baseline information. What this does is it affects the causal relationships, the actual relationships you're trying to study, the drug effects. And this is actually an effect on internal validity. Even the relationships you estimate within your study are wrong, let alone that they don't generalize to a larger population. So here's a classic example of this from an old study, the Coronary Drug Project, that's often used to illustrate this point. So how adherence itself may be related to the outcome. So here the drug clofibrate is being compared to placebo for a mortality outcome. So on the bottom here, we have the percent of people, the percent of mortalities in the clofibrate group, which is about 18%, and the percent in the placebo group, which is about 19%. So there's not much difference here. It doesn't look like clofibrate has much effect on this population on mortality. But there's actually more to this table than I showed you initially. This table also breaks down the results by your adherence, whether you have less than 80% adherence or greater than 80% adherence. This is basically how much of the drug you took compared to what you were supposed to take. And what we see for the ones with higher adherence, those with greater than 80%, the mortality is down to 15%. And those with less than 80%, the mortality is close to 25%. So if you compare that 15% of the adherers, the people who actually took the drug a reasonable amount of time, to the placebo amount, it looks like the drug has an effect. But the thing to be revealed here is the placebo group has the same pattern. So the people who took placebo more than 80% have lower mortality than the people who took placebo less than 80%. Now, obviously, taking placebo more is not going to affect your mortality. What's really being demonstrated here is the people who have a higher adherence are different from the people who have lower adherence. They may have more attention to their own health, or there are any other number of differences between the two groups. And this difference, based on a post-baseline factor, can introduce bias, which doesn't show up if you do not divide by adherence, but when you start looking at adherence, you see that there are differences. So let's talk about adherence and missing data and biases. Patients who complete therapy may be different from those who stop therapy. I think we could think of several reasons for that. The people who complete therapy, again, may be more interested in their health. They may be less sick. They may have less side effects. So there's a difference. So treated group patients who complete therapy may be different from control patients who complete therapy. Now this is very subtle, and you might not be able to pick it up in the time I have on this slide, but the first bullet talks about it, those who complete therapy are different from those who stop therapy. But this difference actually may be different between the two treatment groups. So if the first bullet was the only problem, then maybe you would just look at people who adhere to the drug. Like in the last example, those with greater than 80% adherence. But the reason people don't adhere may be different for patients on treatment versus those on placebo. For example, maybe this drug has a lot of adverse events associated with it. So the people prone to adverse events stop treatment. But on the placebo, those who stop it may be stopping it. They're not stopping it for adverse events because they're only taking placebo. They're stopping it for other reasons. So this is a rather subtle point, but it explains why we just can't look at adherers. Analyzing only patients who complete therapy, or what we call complete data, will likely produce biased results. So that's the kind of conclusion of all this. So let's talk about the different types of adherence and missing data, because this has implications on how you address them, in the design and analysis. So one type is patients may stop treatment. They may stop treatment, but you may actually be able to follow them up after they stop treatment. So stopping treatment is not the same thing as leaving the study. Leaving the study is the patient is not agreeing to have any more follow-up or has just been lost to follow-up perhaps. So a patient can stop treatment and still be followed up. So I'd like to distinguish those two situations. And a third situation is, patients may miss a single study visit. That visit might have involved a particular treatment or a particular assessment, perhaps the endpoint assessment. So you may be missing some of your endpoints. So these are three different types, and it may be important to think of them separately and how you might deal with them. But they're all called intercurrent events, and they should be handled appropriately and not often the same way. So what causes, I've already talked a little bit about this, but what are some of the causes of lack of adherence or missing data? Well, the first thing might be random. So it's not related to anything about the patient's characteristics or the patient outcome. So if this was the only problem, then we could just ignore it, basically. The patients that have the information are just like the patients who don't have the information. It's all random. We could just use the patients where we have the information. We would lose sample size, but we wouldn't create any bias. Another possibility maybe is the frailty of the patient, like sicker patients drop out. And again, maybe it's lack of efficacy. Maybe the patients who are perceiving not getting any benefit from the drug are more likely to drop out. And also I've mentioned side effects. Maybe certain side effects makes patients drop out. Again, it's important to think about this as you design your study and as you come up with analyses and eventually sensitivity analyses. So just a word on adherence. I don't want to make it imply that adherence is forcing the patient to do something. This is not ethical. The protocol should allow for the best interest of the patient. So we're not trying to force adherence. We're trying, the study will represent what happens. We may encourage adherence, through education efforts or whatever. We're not trying to force the patient to do anything that's not in their best interest or not that they're willing to do. So adherence is basically following the protocol, which should allow for the best interest of the patients, even if that means stopping therapy. Okay, so I've already talked about some reasons for missing data, some reasons for lack of adherence. So what are some of the solutions for that? The primary way we analyze efficacy trials typically is intent to treat, which is currently also known as treatment policy. So the first step is we want to get follow-up data regardless of treatment adherence. So even if someone stops treatment, we still like to get the follow-up data. So as I said earlier, stopping treatment is not the same as stopping the study or dropping out of the study. The patient may, once they stop treatment, may not choose to be in the study anymore, and that's their right and choice. But to the extent that patients are willing to continue to be follow-up, even if they're not getting treatment, has an advantage, because we can see what happens to these patients. So we analyze patients according to their random assignment, regardless of adherence and missing data. So this answers the question, what is the effect of being assigned therapy? So you may stop therapy, you may start another therapy, but it does answer the question, if you're randomized to this therapy, what is the effect? And importantly, it maintains the randomization, because you're still comparing people randomized to one group versus the other group. Now, there are problems with the treatment policy or the intent to treat approach. Patients may stop therapy They may cross over to the other group therapy, or they may receive rescue therapy. So stopping therapy is pretty obvious. We already talked about it. In some studies, patients on the placebo group may actually start taking the investigative agent, and that could be because it's available through some other means or it's part of the protocol. Or some protocol often will allow for rescue therapy. If a patient worsens, they don't allow it just to continue to worsen. Some sort of approved rescue therapy is used for that patient. So this creates sort of complications in interpreting this intent to treat, because these things may happen to a patient and may affect your view of like the efficacy of the drug, but are not accounted for in this ITT approach. The ITT approach is basically what it means to, what the effect is to be assigned therapy, not what actually happens to you. And unfortunately, you still may have missing data. So ideally, if a patient stops therapy, we would recommend that you continue to collect data as they would if they were still on therapy, or maybe some abbreviated version, or whatever it takes to get the endpoint you need. But a patient may choose not to continue to be followed up, or they may be lost to follow up. So you still may have missing data. So if to do the intent to treat, you need data on all the individuals. So this is sort of a limitation. So you'll have to find some way of handling this missing data. One approach, not so much for the missing data, but for some of the issues in rescue therapy and stopping, is use a composite approach. So a composite approach would treat stopping therapy, changing therapy, or the use of rescue therapy as a failure in a composite endpoint. So suppose your endpoint was some sort of clinical response, a success or a failure. So the composite would count as a failure not only whatever is being measured, but any of these events like stopping, changing, or rescue would be considered a failure as well. So that's one way to kind of address some of these issues of, that placebo patients may have got rescue therapy, so they're now going to look better. But if you consider that a failure in this composite approach, the ITT approach still works and it still maintains randomization. Again, it doesn't address the missing data, but I'll get to that in a bit. But in general, the approach should be tailored to the specific clinical question you have. Now I'm going to say something about missing data, but mainly use it as an example of sensitivity analysis. Sensitivity analyses involve varying assumptions to explore the potential biases. And the setting we're going to use is the drug effect for patients with missing data is not the same as the drug effect for patients with complete data. I've already said that we don't see bias in the data, but we can use sensitivity analyses to explore assumptions on the bias. This is a rather complicated figure, so I'll spend some time on it. It comes from a study where the primary result was statistically significant, but like all studies, there were missing data. So the green areas in this plot are under certain assumptions where the drug is still statistically significant, and the red areas are where the drug is no longer statistically significant. And there potentially could have been yellow areas where the placebo was actually statistically significant, but that didn't occur. So what we're seeing here is various assumptions of the missing data. The x-axis is assumptions on the estimated mean of the endpoint for those with missing data for the treated group. And likewise, the y-axis, the vertical axis, is assumptions for the estimated mean of the control group with missing data. So most of the patients are not missing data, so that they're still in this analysis. But for the patients that are missing data, we make assumptions of what their mean value would be. And we see different regions where the study is still statistically significant. So this involves clinical interpretation, like what would be reasonable to assume for these missing data. If you can make somewhat extreme assumptions and still see a statistically significant result, then maybe the bias from the missing data is not a big deal. So this involves, again, clinical judgment, but allows you to explore different assumptions for the missing data and maybe see how strong the primary result is. This brings me to concluding statistical principles. In summary, careful design and conduct are needed for reliable results. Randomization addresses baseline biases. Blinding, good adherence, good follow-up, and good endpoint ascertainment addresses post-baseline biases. Pre-specification is important to address multiplicity. Here are my take-home messages, returning to the theme of the beginning of the talk. Address the clinical question, minimize bias and minimize variability, and careful design conduct will promote valid and reliable results. Challenge question number 1. Which of the following does not reduce bias? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. The answer is a larger sample size. If we go back to that example in the unblinded trial with the effort-dependent endpoint of the 6-minute walk and the belief that patients on the experimental treatment may try a little bit harder, on the average, every patient is going to have that 0.5 meters extra effect on the experimental arm, and that bias is not going to go away with larger sample size. Randomization clearly addresses confounding, and blinding clearly addresses endpoint ascertainment, so they do address bias. And I'll leave pre-specification for a moment. The second challenge question is, which of the following addresses multiplicity? A larger sample size, randomization, blinding the knowledge of the drug from the study participants and investigators, pre-specification in the protocol and statistical analysis plan. Here the answer is pre-specification in the protocol and statistical analysis plan. We talked about multiplicity, that if you look at enough things, you're going to see something statistically significant. So pre-specifying what you're primarily interested in will avoid that issue. So with say thank you, and I'm looking forward to answering questions in the question and answer period.
Thanks so much to both Jim and Mark for getting the course started. We've covered a lot of territory in a short amount of time. This is terrific. So we do have some questions that have come through. I'm going to go ahead and start with Jim. The first question, you mentioned that the study population may be narrowly or more broadly defined. Other than enrichment, can you say more about considerations for each of these?
Sure, Kim. Thanks a lot for passing along that question. Yeah, so the study population can be narrowly defined at times or more broadly defined at others. And I think I gave some examples of this, but I'm happy to embellish on it a little bit. So I'm going to think about it from the context of a typical drug development program here, but the principles could apply to other clinical research. So early in drug development, when a drug is first being studied, for example, for a particular use, one is probably doing trials that involve a fairly small number of patients to test a drug, perhaps to explore certain endpoints, to get proof of concept, figure out whether this drug candidate is worth developing further for a particular use. And so really one is trying to identify a signal and really find out whether or not, there's a signal here to carry that drug further in development. And so when we think about how to best define a signal, you can either have a drug with a very large effect, or what's more common is to have a drug that might have a smaller effect, in which case you really want to be able to distinguish it from the noise. And so reducing noise or reducing variability becomes particularly important to try to help identify a signal. And so in early development, one approach to reducing variability is to more narrowly define a patient population such that there is more homogeneity in the population that you're exploring the drug in. And there's obviously limitations to that, and that is you're answering questions about a fairly narrow population that, as the drug is then carried further in development, you might realize that it does not work as well as you hoped once you broaden the population out. But that's one example. Another example could be from a safety perspective. Early in development, when there's not as much known about a particular new drug, you might have very real and legitimate concerns about exploring that drug in patients with certain comorbidities or other complicating features, because you're trying to get not only to protect the patients before you have more safety data accrued, but also it might be more difficult to discern what that drug's safety profile is, on a background of other comorbidities, other concomitant medications and whatnot. But as I mentioned, as a drug goes further along in development, one is not going to want to continue to restrict the population to these fairly narrow groups, but rather really broaden out the population, so that by the time one is putting together an evidence package from both an effectiveness and a safety perspective, the drug has really been studied in the patients for whom you would expect to take the drug if that drug were to be approved. And I would think that the same principles would apply elsewhere in clinical research, that you really want the results from the clinical investigation to be generalizable to whatever the clinical research question that you've asked, and not only applicable to, for example, a very narrow subset of individuals. So hopefully that helps answer the question. Thank you.
Thanks, Jim. And I have one follow-up question that is related. Would the use of enrichment in selecting the study population lead to a, quote, tighter label?
Thanks, Kim. That's a good question too, and you're right, very, very related. So if one were to progress in drug development and use various enrichment strategies, and you generated evidence from clinical investigations in a particularly enriched population, the question is really what scientific conclusions can then be drawn from that, which is really how, if you're seeking drug approval, how labeling would be written, because the labeling should really reflect the use of the drug as it's been demonstrated to be safe and effective. So one example would be, and there's really, I should say, there's no just rules of the road here. Sometimes, and by that I mean that it's very case specific. So, but I'll just give a couple examples that will hopefully illustrate the concept. So let's say one is using a predictive enrichment strategy where you have a drug, as I included in my challenge question, that perhaps has a mechanism that is targeted to a particular, gene variant of a disease. Well, one would then expect for that, the presence or absence of a particular variant to be predictive of whether that drug would work. And if that is in fact the case, then the labeling would very appropriately be limited to the patients for whom you expect the drug to work. And this can be a little bit difficult because it's not always obvious. And sometimes perhaps data are gathered on a biomarker positive, for example, population, and based on very good and solid hypotheses, and you don't have as much data in the biomarker negative population. And perhaps the drug is actually still effective, just less so in biomarker negative patients. And so really the extent to which one actually has data on those populations to see if there could be an interaction between, for example, biomarker positivity and the treatment effect, would all play into decisions about how the label might be written. With respect to prognostic enrichment, it's also not, there's no one-size-fits-all answer. Sometimes it's very reasonable to enrich a trial for a high-risk population because events will happen faster, but there's no scientific reason perhaps to think that the drug would not be effective in patients who are a bit lower risk, maybe moderate risk or whatnot. That maybe the effect size or the absolute risk reduction, for example, would be smaller in the low risk patient. And those are really scientific considerations that one would have to think through. I can think of examples, where prognostic enrichment has led to a labeled indication that is exactly the population that was studied in a trial, and other situations where it was believed that the indication could be broader and not limited to the quote unquote high risk patients, but other sections of the labeling would describe the enrichment strategy so that providers would have that information should they choose to take a look at it. So hopefully that helps, and thanks again.
Great, thanks, Jim. And Mark, we have a couple of questions for you here as well. First, can you say something about what makes a good endpoint in a clinical trial?
Oh, thank you for that question, Kim. That's a very good question. Dr. Jim Smith addressed some of that in his talk, that first and foremost, it should address the clinical question. The endpoint should be chosen to get at what question you're asking for. But there are definitely more considerations that come into play. It should be what we would call sensitive to the drug. So, for example, or sensitive to the perceived effect of the drug. For example, if you're maybe studying an antidepressant and you don't expect to see an effect by week 2, but maybe the mechanism of action or past experience would lead you to believe, you would see effect maybe at week 12, then obviously week 12 is the more appropriate time point for the endpoint. So you're trying to look for something that would be sensitive to the hypothesized drug effect. Now, it still has to be clinically relevant. If reversing that example, if the effect only had an effect on if the drug only had an effect on depression at 1 week and no effect beyond 1 week, well, you might be able to show an effect, but it probably wouldn't be very clinically relevant. So again, the first criteria has to be clinically relevant. But moving on from that, being sensitive to the hypothesized drug effect is important, and then the bias and variability come into play. As I said, you're always trying to minimize bias and minimize variability. Here, variability would mean, like, if for some reason you were theoretically able to take the measurement twice, would you get nearly the same result, or is it going to be very variable? Some endpoints are just naturally going to be not very variable. Maybe a heart attack is a very hard endpoint, and how you measure it, if you were to go back and measure it, take the labs and EKGs, you would get the same answer. Other endpoints may be more variable, endpoints based on maybe a patient-reported endpoint might be more variable. If the patient took the same answered the same questions again, they might come out a little differently. But again, you have to foremost stick with the clinical relevance. So variability is important, but you don't want to answer a question that's no longer relevant. Just one kind of closing thoughts on this is FDA does have a series of guidances and draft guidances on patient-focused drug development. They're mainly focused on the issues of these, maybe something like a patient-reported outcome, although there's other examples in those as well. But they do go into a lot of what makes a good endpoint, both from a clinical relevance point of view and including bias and variability. So I hope that kind of gets at some of at least my thoughts on what makes a good clinical endpoint.
Great, thanks, Mark. And one additional question for you. If a study is not statistically significant, what can we conclude? Can we say that the drug has no effect?
Okay, another good question. I mean, this is a question that goes beyond even clinical trials, but I think it's an important thing to consider. So the concept of statistical significance is a tricky concept, and that's unfortunate. That's unfortunate for you non-statisticians, and it's unfortunate for a statistician. It means, as I tried to explain in the talk, that you've observed something that's very unlikely if the drug had no effect. So therefore, you conclude it probably did have an effect. So when you have a statistically significant effect, you can generally conclude the drug has an effect. I mean, of course, you have to consider all other issues of biases that may have resulted in this. But let's assume it was a very clean, well-conducted, simple trial, and you see a statistically significant effect. Great, you see effects, so you say the drug has an effect. Now, the converse is more tricky. If something is not statistically significant, you cannot automatically conclude that the drug doesn't have an effect. There are several things that may come into play here. Maybe the most important to consider is what I tried to get at is statistical power. It may be that the study had very little power to detect an effect. So even if there was an effect there, the power to detect it might have been very low. The power is a number, is a probability itself. So it can go from 0 to 100%, although it probably can't be either 0 or 100. But typically we design clinical trials to have 80 or 90% power for a given effect. So if we think the effect, say, is 2, whatever that might mean, we have 80% or 90% chance of detecting that. But if we run small trials or trials that are underpowered, we may not detect real effects. So the lack of statistical significance doesn't necessarily imply that there's no effect. Now, if the trial had a lot of power, that's already giving you evidence that if you didn't see it, then it probably doesn't have an effect. But maybe a more straightforward way to examine that is to look at confidence intervals, which I didn't go into in my presentation. But a confidence interval gives you the range of likely effects. And at the upper end of the likely effects is still a very small effect, then you might conclude that the drug doesn't have much of an effect. So again, I hope that addresses this question. Thank you.
Yes, thanks, Mark. And I think we may have enough time for maybe one more question for each of you. Let's go back to Jim. Is placebo the same as no treatment? If not, what's the difference?
Thank you for giving me the chance to clarify this. So absolutely true that placebo and no treatment both mean that someone is not getting an active drug. The difference between a placebo-controlled trial and a no treatment controlled trial is really blinding. So in a placebo-controlled trial, as I described, and I think as folks are probably well familiar, that is going to be a blinded trial because otherwise you're giving participants a placebo and telling them, hey, I'm giving you a placebo, that might not be the effect that you're intending to have, and could introduce bias into the trial. So a no treatment control trial is really when blinding is not possible, and it's going to be an open label trial anyway. Then you still have a control arm that's just getting standard of care, and then your interventional arm is likely getting standard of care plus the open label intervention. So hopefully that clarifies. Thanks.
Great, thanks, Jim. And Mark, one more question for you. How can data fudging, that's a quote, be avoided, and how can you detect if it happens?
Okay, another good question. And I think during my talk, I probably gave the primary way of avoiding it, but I'll go deeper into this. That to avoid data fudging is really a problem multiplicity. I assume that's what's meant here. I assume it's not meant like making up numbers or so. I think what's meant here is that you've looked at a lot of results, maybe you've conducted a lot of studies, and just like my example, you found one subgroup perhaps that was statistically significant, and then you write a paper on that, and you don't mention all the other things you looked at, and you just mention, I have a statistically significant result. That's what I think is meant by fudging here. And as I said in the talk, the best way to avoid that is pre-specification, that you have a protocol in place, and hopefully in a statistical analysis plan if the analysis is non-trivial. And this way, if you set out ahead of time what you're most interested in, and if you find that, it's an important result. And if you don't, that might be interesting to report as well. But if you start reporting on other things that you didn't set out to look at, you have to come clean about that this is an I think Dr. Jim Smith went into this. This is an exploratory endpoint. Although the primary endpoints were not statistically significant, or maybe they were, this exploratory result might be hypothesis generating that we might want to explore in further studies. Now in practice, this is a group for clinical trials. I think it's relatively straightforward. I think there may be requirements for publicly registering your protocol ahead of time, so the public knows what you're looking at. I'm not personally familiar with those requirements, but that's one way to be clear what you're looking at. I mean, you could run a lot of studies and not tell people you're running a lot of studies, and then just pick out the one that's significant. That would also be kind of an issue with fudging, even if you had protocols. I think it's an unlikely scenario for clinical trials because of they're expensive and time-consuming to run. You're not running a lot of trials and just picking out the one that looks best. But I did point out for observational studies, you might see a lot more of that. Now, this group might not be generally focused on that, but it's something to be aware of from kind of a scientific point of view. So thank you. Thanks for the question.
Thank you, Mark, and thank you to Jim also, both for your presentations and for your willingness to engage with the audience with questions. And thank you to the audience for your attention and for submitting some questions for us. We are going to actually go on our first break. And we will resume presentations at 1:45 p.m. Eastern Time. Thank you all.
Welcome back, everybody. I hope you had time to refresh yourselves. So this morning we had a very intense session where we discussed the design and the analysis of clinical trials. And I'm sure you all noticed that the emphasis was very much on evaluating the efficacy of drugs, determining whether they had the effect they were intended to have. We did not talk about safety, and this brings us to our second session. Obviously, safety of the drug products that come through our purview is a fundamental issue, a fundamental responsibility of FDA. Safety considerations change as we move through the drug development process. Early in the drug development process, we're concerned about the safety of trial participants. Later on, obviously, the concern becomes that of the general population exposed to the drug. The other thing that we didn't talk about this morning is that different groups in the population may respond differently to drugs. And that's another area which we're going to touch on in this session, which is special populations or subpopulations that need special consideration. So I'm delighted to welcome our speakers for this afternoon session. Shabnam Nasir, who will talk about the safety considerations in clinical drug development, and she'll be followed by Lin Yao, who will be talking about specific populations in clinical trials. So at this point, let me hand it over to Shabnam.
Hi, good afternoon, everyone. My name is Shabnam Nasir. As was stated, thank you for the kind introduction. I'm a lead physician in the Division of Anti-Infectives in the Office of Infectious Diseases. And my talk today will be focused on safety considerations in clinical drug development. I do appreciate having the opportunity to speak today. So our learning objectives include the following. We'd like to understand that protection of subjects should always be the first priority when designing early clinical studies. We'd like to learn key safety considerations in the conduct of phase 1 trials, and to understand the principles of safety monitoring and reporting in clinical trials. So the main objectives of phase 1 trials are to assess safety and tolerability of the drug product, attempt to characterize dose-limiting adverse reactions, determine maximum dose associated with an acceptable safety profile, to characterize pharmacokinetic parameters, and explore drug metabolism and drug interactions. The subjects in phase 1 trials generally tend to be healthy volunteers, and these volunteers have few confounding factors. In some instances, phase 1 trials are conducted in patients, and these are generally when the drug is known or expected to be toxic. An example would be a trial that's evaluating a cytotoxic agent. When patients are enrolled in phase 1 trials, there will be more confounding factors and sometimes more difficulty in trying to separate disease-related manifestations from adverse reactions. In addition, special populations such as patients with renal or hepatic impairment can also be enrolled in phase 1 trials. So here are some general considerations when one is moving from the non-clinical to first-in-human study. So we have to consider what is the evidence we've garnered so far from the non-clinical studies. And so we have to ask ourselves a number of questions. What is the duration and the total exposure proposed in humans? And how does that correlate with what was tested in the animals? We have to think about the characteristics of the test drug. Is it a small molecule? Is it a biologic? Does it have a long half-life? We have to consider the disease that's being targeted for treatment, because that would need to be taken into consideration in the benefit and risk assessment. We have to think about the population in which the drug will be used. Is the intent to enroll women of childbearing potential or children in the study? And what's the route of administration? Is the product being given systemically or topically? So these are some of the initial considerations. Do the non-clinical studies that have been done so far provide sufficient data to allow for the proposed clinical trial to be undertaken? So what species were they studied in? Are the species appropriate and are they relevant to the particular drug? What are the potential target organs of toxicity? What was the duration studied in animals, and what was the dose and the route of exposure? Was it appropriate and similar to what is being proposed in the human study? And what about the PK and PD assessments? Were they appropriate? Was a dose response identified? And then again, do we need to do any special studies in animals if we're planning to enroll, for example, children or pregnant women? So it's important to keep in mind that only some toxicities that are seen in non-clinical studies translate into adverse reactions in humans, while there's other toxicities that do not. Excuse me. And so you end up with both predictable and unpredictable toxicities, and these can appear in any phase of development, and sometimes you actually only see them in the post-marketing setting. It really depends on the size of your safety database that was available pre-market. It's also important to keep in mind that certain adverse reactions are subjective, and these can be adverse reactions such as headache or other events such as hypersensitivity reactions that really can't be assessed in the non-clinical arena. So let's talk about some examples. So here's an example of predictable toxicity. And this example is from the anti-infective space where I'm from. I'm sure you can find other examples in other therapeutic areas. So linezolid is an antibacterial drug that was approved in the late 1990s. And at that time, it was the first member of the oxalidenone class. It had activity against gram-positive organisms, and was also thought to have activity against some resistant organisms. So certainly there was some therapeutic benefit that could be offered by the product. The non-clinical studies that had been conducted showed that myelosuppression occurred, and this appeared to be both dose and time dependent. There were various changes in the bone marrow, and it appeared to affect all 3 cell lines. But the product had the potential to offer therapeutic benefit, and so further clinical development was pursued. When phase 3 trials were conducted, there was no evidence of myelosuppression per se, but there was a high frequency of thrombocytopenia that was seen in the linezolid-treated patients. And at the time the product was approved, the prescribing information included precautions for thrombocytopenia, and they also noted a clinical the animal pharmacology section described the hematopoietic effect noted in animals. After the product was approved, there were cases of myelosuppression seen affecting all 3 cell lines, and these were reported as leukopenia, anemia, etc. And so labeling was updated to include a warning regarding myelosuppression. Here's an example of unpredictable toxicity. So we had 2 products, both were members of the beta-lactam class, which is a very well-known class of antibacterial drugs, and we have a lot of experience in this class. The structure of these beta-lactams, however, had been modified to enhance the spectrum of activity. There were no unexpected toxicities seen in animals, so both of the sponsors proceeded to conduct their phase 1 studies. The single dose went well, but then when they went into the multiple dose trial, the subjects developed moderate to severe skin reactions such that the product development actually had to be halted for both products. So in this case, this is an example of unpredictable toxicity, and even if the non-clinical studies appear benign and you don't see any adverse events, it doesn't mean that you're not going to see events when you conduct the study in humans. Now to a discussion on maximum recommended starting dose. And you will be hearing more about this in day 2 lectures, but I'll just go over some high-level concepts from the clinical perspective. So There are some key principles in selecting an MRSD. One is that you want to avoid toxicity at the initial clinical dose. But at the same time, you want to allow reasonably rapid attainment of the trial objectives. And these are usually tolerability and PK, so that you're not so low that it'll take you forever to get where you need to be, where the target dose was. And typically, we use an algorithmic approach based on what doses were given to animals and what toxicities were observed. There are some alternate approaches based on animal pharmacokinetics and modeling, but for the most part, we use the algorithmic approach. Some of the key concepts when you look at the MRSD are the no observed adverse effect level, or the NOEL, and this is the highest dose tested in animal species that did not produce a significant increase in adverse effects compared to a control group. The second term is HED, or human equivalent dose, and this is really what is the dose in animals that converts to a certain milligram per kilogram dose in humans. Based on that, you would be able to come up with the starting dose. Then you also have to consider which animal species you use to make these calculations. Ideally, you would choose the most sensitive species, and that's the species from which you can identify the lowest HED. In some cases, however, you would want to use the most appropriate animal species because that species actually might have the particular target of interest, and this is especially seen with biologics. So here's a schematic that shows the steps in determining the maximum recommended starting dose, and for your reference, the guidance is at the bottom of the slide. So first you would determine the NOEL, which is a starting point for determining a safe starting dose for new molecular entities in healthy volunteers. Then that gets converted into the HED. You select the HED from the most sensitive or appropriate species, and then you choose a safety factor. Then you divide the HED by that safety factor to come up with a maximum recommended starting dose. So what is the safety factor? It's a number that gives you a margin of safety for protection of human subjects when you're moving from animal studies to humans receiving the initial dose. The default number is 10, but there is some degree of flexibility, and sometimes we can go higher or lower than 10. The safety factor provides us some reassurance that when we're moving from animals to humans, that we're not exposing human subjects to any great risk. One knows that there's variability when you move from animals to humans, and the safety factor allows for that variability to be accounted for. So, as I mentioned previously in the examples, there are uncertainties when you go from animals to humans. There can be differences in the sensitivity between animals and humans to certain products. There can be difficulty in detecting certain toxicities in animals, such as headache, myalgia. There could be differences in receptor densities or affinities. You always have to be prepared for unexpected toxicities. In addition, there could be interspecies differences in how the product is handled, such as ADME. And keep in mind that, again, humans and animals may not always handle the product in an identical manner. So when would we have to increase the safety factor greater than 10? If it's a brand new class and we have no prior experience with the product, you might have to consider increasing the safety factor. Other issues could be that the toxicities in the non-clinical studies were severe or irreversible, or they could be toxicities that are non-monitorables. For instance, if you have histopathologic changes, but they were not monitored because there's no clinical methods to monitor those toxicities, or there are no biomarkers, that could be a reason to consider increasing safety factor. If there's a very steep dose response curve, that could indicate a greater risk in humans. If the PK is not linear, it limits the ability to predict dose-related toxicity. Also, if there's variable bioavailability, because you could have poor bioavailability in animals, and that may underestimate the toxicity when you move to humans. On the other hand, you could decrease the safety factor to less than 10, and some of the reasons would be if you're dealing with a member of a well-characterized class of drugs, if the toxicity is easily, that's produced is easily monitored, if it's reversible, if it's predictable, and also if the NOEL was determined based on toxicity studies of a longer duration, you might have a little bit of more reassurance in reducing the safety factor. So let's discuss an example of how you would calculate the MRSD. Here's a product for which the HED was about 400, derived from rats. The sponsor proposed a starting dose of 100 milligrams, so that brought the safety factor to 4. And the rationale provided was that this was a member of a well-characterized class of drugs, and that the toxicity studies in 2 species were of longer duration than what was proposed in the clinical trials. In addition, the potential toxicities were readily monitorable and reversible. However, we actually didn't agree totally with the rationale because we knew that members of the class had exhibited more toxicity than the parent class from which it was derived. And there was also some concern that the bioavailability in animals was low and that human bioavailability could potentially be greater than anticipated, leading to a risk of higher exposure in humans. So we actually agreed upon a starting dose of 50 milligrams, raising the safety factor to 8. So now let's move into some general considerations when you're going to be conducting these phase 1 trials. So more questions to ask yourself when you're getting ready to conduct these trials. One is, are the clinical trial protocols designed appropriately to ensure safety and meet the stated objectives? You have to protect the safety of the subjects in the trial as always. And do you have enough information about the quality of the investigational products? For example, has the formulation been well characterized? Do we know, for example, what the impurities are? Are the route and the rate of administration appropriate? Can the product be given as a bolus dose, or are there toxicities such that we would have to give it as a slow infusion? What is the mechanism or mode of action? Is it novel? Do we know anything about similar products and that act via a similar mechanism? Are you expecting an off-target effect? And that's a very important concept, especially if you're dealing with a biologic and the mode of action involves a target which is connected to multiple signaling pathways or affects biological cascade or cytokine release. Let's go into safety considerations regarding dosing. How do we determine an appropriate dose? Ideally, one subject should receive a single dose, and that would be followed by sequential administration within the cohort. You have to ensure that there is an adequate period of observation between dosing so that one has the time to observe and interpret adverse reactions in the cohorts at the lower dose. And how long you actually observe will depend on the properties of the drugs, the PK properties, how long it stays in the system, how long its half-life is. And so we also learn good lessons from previous trials conducted with similar products. So moving on to dose escalation. If the protocol for these phase 1 trials allows for dose escalation, you have to ask whether the dose escalation scheme is appropriate. Are the dose increments appropriate? Is it okay to double up or go up by 4 times? You have to be cautious and use a slower rate of dose escalation, especially if you have a small therapeutic window, if you had animal models that were not very good, or if you had any concerns about toxicity. There is a fair bit of flexibility here. Again, it just depends on your product, the target, and the toxicity found in animals. You have to ask yourself if the amount of information before each dose escalation is appropriate, and also if the number of subjects at each dose is appropriate. So next we'll move to safety monitoring. It's important that protocols have an appropriate monitoring scheme to look at clinical signs and symptoms of adverse events likely to be associated with the drug. And the duration of the clinical observation should also be adequate with respect to the stated objectives and endpoints, the anticipated response to the product, and the health-related conditions that are being studied. Sometimes we do need to monitor for a longer period of time, and again, it depends on the characteristics of the drug. Where we monitor also matters, whether it's a hospital setting that's needed. And if you're worried about any sort of delayed onset reactions, undetected toxicities, you have to make sure that the follow-up is long enough. So as far as frequency of monitoring, there may be a need to monitor more frequently soon after initial dosing in the first week or so. Certainly you would need to have follow-up visits for subjects who develop adverse events or laboratory abnormalities. In addition to monitoring clinically for adverse reactions, you'd also monitor laboratory tests. And then which particular laboratory tests you order and collect the data on really depends again on your drug and the target organs of toxicity. You want to ask yourself if the laboratory tests would include routine assessment of all organ systems. Do we know enough about the drug such that you only need to assess specific organ systems? Are they sufficiently detailed laboratory tests that you can actually make an assessment of whether or not the drug is causing toxicity of those specific organs? And then lastly, are there stopping rules for patients whose laboratory test abnormalities reach a certain threshold? So what are stopping rules? It's important that the protocol have clear stopping rules for administering the drug, enrollment, dose escalation, and rules for termination of the trial itself. Oftentimes, we've actually had to put INDs on hold because there were no stopping rules in the protocol. So again, these are changes to the protocol that are to be implemented when toxicity is observed. And to be able to have stopping rules, you have to identify what might be acceptable toxicities and what are not acceptable toxicities. Since it's unlikely that you'll have a drug with no toxicities, it's important to stop and try to generate these stopping rules. If acceptable toxicities occur, these will be the ones that don't result in changes in how the trial is continued in terms of subject enrollment and dosing. If you have other toxicities that are not acceptable, you have to have a procedure in place for what you would do with the study. So let's go on to our challenge question 1. Which of the following is used to calculate the MRSD? And hopefully everybody will get this. And this is the Noel. All right, so let's move on to sort of the second half of my talk, where we talk about where we continue to talk about the evaluation of safety, but sort of in larger, later clinical settings. So safety evaluation is a central component in all stages of drug development. It's an evolving process. It depends on the stage of the product under development. For products that are marketed, the safety information is reflected in the product labeling. It's also called the prescribing information, or PI. While up-to-date safety information for products that are under investigation would be contained in the Investigator brochure, or IB. So again, these would be non-clinical findings from in vitro or animal studies, clinical pharmacokinetic safety data. And for unapproved products, the investigator's brochure is equivalent to the prescribing information. So what are the sources of safety information found in the investigator brochure or the prescribing information? Non-clinical data, again, can include in vitro or animal data, as well as information about the structure of the product and the manufacturing process. Clinical pharmacological data can include information about PK/PD, And then safety data can come from clinical safety information from healthy volunteers and patients. Perhaps the most pertinent information, of course, will be the data from the clinical trials for the same indication, as these data are generally controlled for the patient population characteristics and the underlying pathological process, and also from trials for different indications. You can also have information in the IB from post-marketing experience and, IB or prescribing information from post-marketing experience, the published medical literature, and last but not least, safety profiles of drugs in the same class. So now, why is safety monitoring required in all clinical trials? The goal of safety monitoring in clinical trials is to identify, evaluate, minimize, and appropriately manage risk. And this will lead to our discussion of adverse event ascertainment and monitoring. So we have a number of regulatory definitions that we'd like to go over, and I have given you the CFRs for your reference. Adverse event is any untoward medical occurrence associated with the use of a drug in humans, whether or not it is considered to be drug-related. This could be an unfavorable sign, symptom, disease, abnormal lab, a vital sign, or other testing, or a worsening of the above or a constellation of the above. Adverse events can also be referred to as adverse experience. Some examples of ways that adverse events can be ascertained include spontaneously reported or observed signs and symptoms. Symptoms are signs that are reported as a result of a probe, such as a questionnaire or a checklist. And then testing of vital signs, laboratory studies, and special safety assessments can also be conducted, and these can include, for example, visual, hearing tests, or EKGs. Upon reporting, adverse events are characterized according to their severity, and there are a number of severity grading scales or tables that are available to provide guidance to sponsors or investigators for safety monitoring in clinical trials. These are optional tools. They are specific to the study population that's being evaluated, for example, patients or healthy volunteers. Clinical abnormalities may be represented as either mild, moderate, severe, potentially life-threatening, for example, in healthy volunteers. If you have oncology patients, AEs could be graded by a scale of 1 through 5, where 1 is mild symptoms to grade 5, which is death related to an AE. So again, they're specific to the study population. And it's important to keep in mind also that here are some examples. There's probably more. So again, it's important to keep in mind that the term severe is not the same as serious. What happens when an AE report gets to the sponsor? So the process includes conversion from the investigator's verbatim terms to the standard preferred terms using a specialized dictionary called MEDRA. MEDRA is the Medical Dictionary for Regulatory Activities, and it can be used to sort AEs and group-like events. Terms are used to calculate the incidence of AE. Here's a schematic to illustrate the MEDRA hierarchy. It starts with the highest level of terminology, and that's the least specific, which is the systemic organ class, or SOC. And you move down all the way to the lowest level terms. This is in order of increasing specificity. The PT represents a single specific medical concept. So clinical investigators play a really crucial role in the safety reporting, and you would want them to use the most scientific and consistent terminology when reporting adverse events, as coding problems could result in splitting or lumping adverse events and therefore missing safety signals during the safety data analysis. So splitting similar terms such as hypertension, high blood pressure, these can result in a lower incidence, or they could minimize or mask a safety signal. And on the other hand, lumping dissimilar terms to the same PT could obscure a safety signal under a lumped term. So for example, facial edema is very different from leg edema as far as pathophysiology, and a coding issue could be lumping all edema to the PT edema. In this third bullet, there could be a lack of an adequate term or definition to represent a syndrome of interest such as drug hypersensitivity or serotonin syndrome. And in this case, you may need a prospective case definition if the syndrome is not well characterized by a single term. Moving on to another regulatory definition of serious, again, not to be confused with severe. Serious adverse events are any adverse events that, in the opinion of the investigator or sponsor, results in the following. So death or life-threatening conditions with the immediate risk of death, in hospitalization or prolongation of existing hospitalization, disability or a birth defect, and last but not least, important medical events. And these would be those that could jeopardize the patient or require intervention to prevent one of the outcomes listed above. Examples may be allergic bronchospasm, which would require intensive treatment in an ER or at home. So here's a list of examples of uncommon SAEs. And if you see one of these, it could raise a red flag that there might be a possibility that the AE was caused by the study drug, especially if you have a study population of generally healthy individuals. You really shouldn't see one of these SAEs. This is not a comprehensive list, but it may be a good start. So on this slide, I'll discuss points to consider for investigators in evaluation of SAEs. SAEs are, like any other adverse event, need to be assessed for drug-related causality. And you can do that by asking a number of questions. One would be, is it common? Is it a common occurrence in the population under study? Was it treatment emergent, meaning did it occur on treatment or shortly thereafter? Did it respond to drug discontinuation or de-challenge? Did it reoccur upon introduction of the study drug or re-challenge? Were there concomitant medications? Were there any pertinent laboratory studies or other tests done to try to characterize the adverse event further? Was there an obvious alternative cause? And lastly, but not least, was the SAE a study endpoint? For example, if death occurred, was that also a study endpoint? So you also want to report adverse events by providing enough relevant clinical information in the case report form. You want to include enough information in the case report form to allow a good quality narrative. So now let's talk about what the clinical investigator reports to the sponsor. And again, the CFR is there for your reference. So an investigator must immediately report all SAEs, whether or not the event is considered drug-related. The investigator must also include an assessment of whether there's a reasonable possibility that the drug caused the event, and this is usually called a causality assessment. Study endpoints that are SAEs must be reported in accordance with the protocol. One exception is that if there's evidence suggesting a causal relationship between the drug and the event, then in that case, the investigator must immediately report the SAE to the sponsor. In addition, the investigator should also record non-serious SAEs and report them to the sponsor according to the timetable that's noted in the protocol. So let's discuss a hypothetical case. I think that this might illustrate some of the points mentioned on the last couple of slides. This is case 1. You're an investigator for a clinical trial evaluating whether antihypertensive drug A is associated with a reduced risk of death, MI, or stroke, and death is a study endpoint in the trial. You're told that an 80-year-old white male just died in the trial, and we're also actually told the cause of death was anaphylaxis. So the question is, do you have to immediately report this case to the sponsor? So let's take a minute to think about what to do here. So you're told that death is a study endpoint, and usually an SAE that is also a study endpoint can just be reported per protocol. However, here we're told that the death was due to anaphylaxis. So if you recall in the previous slides on uncommon SAEs, anaphylaxis was listed, and with that SAE, that should be sort of a red flag that there could be evidence to suggest a causal relationship between drug A and event, and so the investigator must immediately report this event to the sponsor. So the question is yes. The answer is yes to this question. All right, so moving back to some regulatory definitions, we'll discuss the ones important to expedited reporting of adverse events to FDA. And the concept of unexpected adverse event is presented here. An adverse event is deemed unexpected if it's not listed in the investigator brochure or IB, or if the IB is not available or required, if it's not consistent with the risk information described in the general investigational plan or elsewhere in the current application. It would be unexpected if it's listed in the IB, but it's not at the specificity or the severity observed. For example, the IB lists increased creatinine, but you have a case of acute renal failure, and that would be considered unexpected by virtue of greater severity. Other examples would be if you have a cerebral thromboembolism, and this would be considered unexpected by virtue of greater specificity if the IB listed only CVA or cerebrovascular accident. And it could also be considered unexpected if it's mentioned in the IB as anticipated due to the pharmacological properties of the drug, or if it occurs with other drugs in the class, but not with the particular drug under investigation. So one example would be angioedema. It's anticipated to occur in patients that are exposed to the class of ACE inhibitor drugs. Angioedema would be described in the IB as a class effect, but a case of angioedema observed with the drug under investigation would still be considered unexpected for reporting purposes until it's included in the IB as occurring with the drug under investigation. Go on to another hypothetical case, case 2. Here you're the clinical investigator for a trial evaluating a new anti-malarial drug B for the treatment of acute uncomplicated malaria. The investigator brochure lists elevated hepatic enzymes. And the question would an event of hepatic necrosis in this trial for drug B be considered an unexpected AE? So I'll give you a moment to think. So the answer is yes, hepatic necrosis in this hypothetical case would be considered unexpected. And the reason is because the IB does not list the AE at the severity observed. So you're seeing hepatic necrosis when only elevated hepatic enzymes are noted in the investigator's brochure. Okay, so moving on to our next definition, suspected adverse reactions. So a suspected adverse reaction is an adverse event for which there's a reasonable possibility that the drug caused the adverse event. Again, reasonable possibility means that there's evidence to suggest a causal relationship between the drug and the AE for purposes of IND safety reporting. So let's look at some examples of suspected adverse reaction. This could be a single occurrence of an uncommon event that is known to be strongly associated with drug exposure. So for example, angioedema, Stevens-Johnson syndrome. Another example would be one or more occurrence of an event that's not commonly associated with drug exposure, but is otherwise uncommon in the exposed population. So if you have healthy subjects and they're developing neutropenia, that could be an example of this, or a heart valve lesion in a young adult, or tendon rupture is another example. And another type of example is an aggregate analysis of specific events observed in a clinical trial, indicating that those events occur more frequently in the drug treatment group than in a concurrent or historical control group. And an example of this would maybe having several acute myocardial infarctions in long duration trials of elderly cancer patients. So overall, the determination of an AE as a suspected adverse reaction it can be difficult, but it's really critical to avoid submission of uninformative safety reports. And the sponsor is the one that should evaluate all available information and decide whether there's a reasonable possibility that the drug caused the AE. And that's because the sponsor is actually the ultimate party that determines if a drug is or is not causally related to the AE. Here I wanted to show a slide that illustrates the universe of suspected adverse reactions. So these are actually not just adverse events with undetermined causality, and adverse events here are noted in the outermost yellow sphere, but they're not yet definitely established adverse reactions, which are noted in the dark red sphere in the center. So suspected adverse reactions are actually in the middle. Adverse reactions are a subset of all suspected adverse reactions where there's a reason to conclude that the drug caused the event. Now let's talk about IND safety reporting by the sponsor to the FDA. FDA continues to require that 3 criteria generally be met before a sponsor submits an IND safety report. And again, the CFR is provided here for your reference. So namely, the event must be serious, unexpected, and there must be evidence to suggest a causal relationship between the drug and the event. Often this standard is called a SUSAR, and it stands for Serious and Unexpected Suspected Adverse Reaction. So again, the sponsor must assess whether or not there's a reasonable possibility that the drug caused the event, and the sponsor should only submit these type of IND safety reports when all 3 criteria are met. In addition, sponsors are expected to submit an annual report in the IND that includes a summary of the most frequent adverse events. It should also contain a summary of the SAEs. So now let's talk about timeframes when it comes to IND safety reports. These reports are required to be submitted to FDA in 15 days, and those that are required to be submitted within 7 days. As you can imagine, the most serious events must be reported within 7 days. And there are 5 categories of adverse events that trigger the sponsor's responsibility to submit an IND safety report. One is the SUSAR that we just mentioned, serious and unexpected suspected adverse reactions. Findings from other studies suggesting a significant risk in humans. Findings from animal and in vitro testing suggesting a significant risk in humans. An increased rate of occurrence of serious suspected adverse reactions, and serious adverse events from bioavailability and bioequivalence studies not under IND. And sponsors have to submit IND safety reports within 15 days of the adverse reaction unless the adverse reaction was fatal or life-threatening, in which case the report must be submitted to FDA within 7 days. And there may be additional details that you might want to look at in the guidance that I've provided in my references regarding the timeframes. So in summary, I've tried to provide a high-level overview of safety considerations in phase 1 trials. Important questions to ask yourself before you decide to dose humans as you translate the information from non-clinical studies to either healthy volunteers or patients. what's the relevance of toxicities seen in the non-clinical studies, and how they translate to adverse events in humans. It's important to keep in mind that there are both predictable and unpredictable adverse reactions. We also discussed how to go about determining a safe starting dose in humans and the concept of the safety factor. We next discussed the importance of safety monitoring and stopping rules, and the ascertainment of safety in larger clinical trials, and some important regulatory definitions of adverse events and monitoring. Finally, we also discussed how investigators play a really integral role in assuring quality safety assessments and the requirements of reporting to the sponsor. And then in turn, we discussed about how the sponsor will evaluate all available safety information and report to the FDA and all participating investigators. Okay, so this brings us to our last challenge question. This is a true or false question. If the adverse event meets all three of the definitions for suspected adverse reaction, serious and unexpected, it should be submitted as an IND safety report. So I'm going to give you a few seconds to think about it. So the answer here is true. I hope everyone got this one. All three criteria should be met for it to be submitted as an IND safety report. Now I just wanted to show you my references. If you had any more questions, please refer to these. And I thank you so much for your time today. Appreciate the opportunity to speak. And the next speaker will be Dr. Lin Yao, who will present on special populations. Thank you very much.
Hello, everyone. I'm delighted to be here today to give you some of my perspectives on specific populations in clinical trials. Here are the objectives of my talk. I'll be focusing on drug development considerations for children and pregnant and lactating individuals, and I'll mention some of the work we're doing currently in enhancing diversity in clinical trials. So FDA labeling regulation includes requirements for labeling of specific populations, and you can see here some of the specific populations that I'll be talking about today. There are other specific populations that I will not be discussing, including geriatric populations and patients with renal or hepatic impairment. So let me start this section with describing the historical problem for children. Prior to about the late 1990s, very few drugs that were used in children were actually approved for use in children, which of course led to significant off-label use. Why was this the case? Well, first of all, we understand clearly that there are differences between children with respect to development, drug metabolism, and toxicity, potential toxicity issues that require that we study drugs in children if we're going to use them in children. But often those studies were discouraged because of many issues, including ethical issues, fear of harming children, and of course perceived liability. There was also a lack of incentive for drug companies to conduct studies. And what this all led to was a situation in which practitioners were faced with a choice, either to not treat children with potentially beneficial medicines because they were not approved for use, or to treat patients based on limited data, anecdotal experience, or based off information from adult studies. In the late 1990s and early 2000s, there became a clear recognition that children should have access to products that have been appropriately evaluated, and that when drug developers are developing drugs for an adult use, if that drug would have a potential for use in children, that those product development programs should include pediatric studies. And of course, there's an important regulatory standard that must be met for conducting pediatric research. And these regulatory standards increase the strength and the quality of the research. These concepts were all outlined in a very important document, the ICH, that's the International Conference on Harmonization, E11 and E11R1, clinical investigations of medicinal products in the pediatric population. Around the same time that the ICH E11 was originally published in 2000, there was a movement in the United States to ensure that children had access to approved drugs as well. And so there were advocates, children advocates across the country who lobbied Congress to pass pediatric drug development laws. And what we had passed in 2002 was the Best Pharmaceuticals for Children Act, or BPCA, and then shortly following in 2003, the Pediatric Research Equity Act, or PREA. PREA, as you can see on the left-hand side, requires studies. So FDA is given the authority to require studies under certain circumstances for diseases or conditions that occur both in adult and pediatric populations. Now, importantly, products with orphan designation are exempt from requirements under PREA, with the exception of certain pediatric cancer drugs, and information from those studies that are required by FDA must be labeled. On the other side, BPCA is a voluntary or incentive provision, which allows for 6 months of additional marketing exclusivity if studies are completed and submitted to FDA that are in response to FDA's request for certain studies. Now, it's also important that orphan designated products can be included under BPCA, and information from those studies must also be included in labeling. Now, you can see that there's an intersection between PREA and BPCA, and we believe that these two laws work together hand in hand to increase the availability of approved drugs and biological products for the treatment of children. So on the next few slides, I want to provide some comments on therapeutics development in children. And importantly, in the US, there's an evidentiary standard for approval, and that approval is no different for children compared to adults. And that is, as you've probably heard from previous talks, that in order for a drug to be approved in the US, there must be demonstration of substantial evidence of effectiveness and clinical benefit. That is, how a treatment impacts a patient's ability to feel, function, or survive, or that it improves the delay in the progression of clinically meaningful aspects of a disease. And that evidence is usually required through the conduct of adequate and well-controlled investigations. And there must also be important safety information included to apply an appropriate risk-benefit analysis. So while the approval standards are the same for children and adults for drugs in the US, there are special considerations that relate to children. First, and very importantly, are the ethical considerations. And I could give a talk for an hour just on the ethical considerations. But the 3 points highlighted here are very important and include that children should only be enrolled in clinical trials if the objectives and the scientific data necessary cannot be met by enrolling a population that can provide informed consent. Also, a child cannot be enrolled in a clinical trial unless there's a prospect of direct benefit. And that's important for any trial that would expose a child to a risk that would be considered any more than a minor increase over minimal risk. And then also, children should not be obviously placed in a disadvantage for enrolling in a clinical trial. There's also important feasibility considerations with respect to pediatric product development. And this is, I guess, a good thing for children, and that is that generally children are healthier, and that the prevalence or incidence of disease in many pediatric populations is lower than that seen in that same disease in adults. Nevertheless, these feasibility considerations can play an important role in successful completion and the design of pediatric clinical trials. So I've described the ethical and feasibility differences in therapeutics development between children and adults. There are additional special considerations that I want to go over on this slide. Firstly, formulation development may be different, and that may seem obvious to many of you, but children below a certain age may not be able to take and swallow safely in a solid oral dosage form, so there may be a need for different types of formulations that are acceptable to children. Additionally, even if you have an appropriate oral dosage form, you may need to consider different strengths or different concentrations to allow for accurate dosing of all pediatric age groups. Animal toxicology studies, there may be a need when there are specific developmental safety concerns to conduct specific juvenile toxicology studies, and I provided a very important guidance, and another ICH guidance, S11, that you can refer to when considering the need for juvenile toxicology studies. There are clearly study design considerations ranging from dose selection to the choice of endpoint or control, as well as the monitoring of safety, including developmental safety issues that should be considered. And then the timing of the studies. When should the pediatric studies be initiated relative to the initiation or the completion of adult drug development? And then finally, and I'll talk later on about the use of pediatric extrapolation, a very important framework to consider, and I'll speak about that in a few minutes. Many of these, or all of these considerations, are discussed, as I mentioned, in that ICH E11 guideline, and then the revision to the guideline, and I've created the link here for you. So now I want to discuss pediatric extrapolation. As I mentioned briefly on the previous slide, this is a very important concept that can be applied specifically in pediatric therapeutics development programs. This new guideline that was published in August of this year, the ICH E11A Pediatric Guideline, goes into very important details on how to apply pediatric extrapolation. But let me just spend a little bit of time to explain how that can be applied in a pediatric development program. First of all, pediatric extrapolation is defined here in the first bullet. But what I really want to explain to you is that in an adult development program, in a successful one, when you've established efficacy and safety, in the subsequent pediatric development program for that same condition, if it is determined that there is sufficient similarity between the disease, the drug pharmacology, and the response to treatment between the adult reference population and the target pediatric population, then you may be able to develop or design a program that can leverage the data already collected on safety and efficacy in the adult program and apply that to the pediatric program. Clearly, if data can be leveraged in an adult program that would support a pediatric program, this could lead to an increase in the efficiency and success of pediatric drug development and greatly decrease the time necessary. However, in order to apply pediatric extrapolation, as described in the guidance, there needs to be robust information to support the similarities between the adult and pediatric populations. And importantly, any differences that may affect the degree to which pediatric extrapolation approaches can be applied. The guidance also describes that efficacy and safety can be extrapolated given the appropriate support and justification. And another important point about developing a pediatric extrapolation approach is the need to have multidisciplinary expertise, including specialists in pediatrics and in the specific disease, clinical pharmacology experts, as well as biostatistical expertise. There are many examples of pediatric extrapolation that I could go into, which I won't, because I think it's better to refer you to this guidance and to read it carefully, because there are great examples within the guidance, as well as training material examples at the ICH website that I can direct you to for more information. So now that I've given you a taste or flavor of the important components of a pediatric drug development plan, here's where I want to talk to you about some regulatory requirements. So under PREA, as I mentioned, the Pediatric Research Equity Act, there's a requirement for sponsors to submit their plans for how they're going to study children that they may be required to study under PREA in the submission to FDA of an initial pediatric study plan, or IPSP. This guidance was finalized in 2020. And in this guidance, it goes through important considerations for what to submit, how to submit, and when to submit these IPSPs to FDA. And this IPSP is really intended to provide FDA with a chance to review plans for pediatric therapeutics development and to provide advice and recommendations about those plans. Importantly, you can get a lot of information from the guidance, and I've provided the link here. But we want these plans submitted as early as possible during drug development, but certainly no later than the end of phase 2 in most situations. So what have been the effects of BPCA and PREA over the last almost 25 years now? And as you can see here, again, prior to the late 1990s, almost no drugs had pediatric-specific labeling. And you can see here that there's been really a clear linear increase in the number of labeling changes with pediatric-specific information over the last 20 years. And I'm happy to report that we have over 1,100 labelings with pediatric-specific information over the last 20 years. So I believe that this is evidence of the effect, the very important positive effect that the laws in the U.S. have led to drugs that are now appropriately labeled for use in children. Now I'd like to turn my attention to inclusion of pregnant individuals in clinical trials. Some important background information. There are approximately 5.5 million pregnancies in the United States each year, and clearly pregnant women may have chronic conditions that exist before they're pregnant, or they develop conditions while they're pregnant, and they can also have conditions specifically related to their pregnancy. Just like children, though, pregnant individuals have historically been excluded from drug development trials. And oftentimes at the time of approval, the only safety information we have come from non-clinical, usually animal reproductive toxicology safety studies. So here on this slide, I want to make some remarks about similarities and differences between adults and children. Specifically pregnant and lactating adults. So children, pregnant, and lactating adults should have access to medicines that have been appropriately evaluated. But when a drug is approved in adults, it's also approved in pregnant and lactating adults, which is unlike the situation in children, where approval in adults does not automatically mean that safety and effectiveness have been established in children. However, in pregnancy and lactation, there are important physiological changes that can impact the safety and dosing of drugs. And oftentimes at the time of approval, the only information we have are from animals. Therefore, there's an important need to consider collection of safety and dosing information in addition to what has already been collected in non-clinical studies. And this is all to support prescribers and patients being able to make informed decisions about whether to take a drug during pregnancy or lactation. As I mentioned, just like children, there has been this general approach to automatically exclude pregnant or lactating individuals from clinical trials. But there's been an increasing awareness over the last few years that there needs to be a paradigm shift, and that there are interested stakeholders and advocates that recognize that pregnant and lactating individuals can be safely included in clinical trials under certain circumstances. There are also regulatory advances that are occurring to change this automatic exclusion paradigm, including that the common rule, which is regulation that is promulgated by NIH to remove the reference of pregnant individuals as vulnerable, and FDA is working to harmonize this regulation with the NIH regulation. So what are some issues that must be considered before including pregnant individuals in clinical trials? I want to point out that FDA published a guidance on this topic it's a draft guidance in 2017, and this information is all contained in the guidance. But let me go over some important highlights. The first is, before pregnant individuals can be studied, there must be at least some margin of safety established through the conduct of non-clinical studies in pregnant animals. And that's no different than going into adult populations or pediatric populations, that we must have the safety identified or evaluated, generally first in non-clinical studies. In the pre-marketing setting, it's important, again, because there is the need to understand the benefit to the pregnant woman and fetus before enrolling a pregnant individual in a clinical trial pre-marketing, that there must be a prospect of direct benefit, and that information that you obtain by enrolling that pregnant individual cannot be obtained by other means. In the post-marketing setting, generally would like to have some safety data available in non-pregnant individuals, adults, and that there may be some information to support it from the literature. Now, in addition to that two conditions exist. One, that efficacy can't be extrapolated, and two, that you can't get the safety information from any other means. And these are important principles that need to be present before enrolling pregnant individuals, either in the pre-market or post-market setting. The other thing that's important that's discussed in the guidance is what happens when a patient becomes pregnant during the clinical trial. And there are important considerations there, and to not lose the opportunity to collect some information because that patient became pregnant during the trial. Now, that patient should be immediately unblinded, so we know whether or not the patient was exposed to drug or whether not through or just placebo, and that the patient should undergo a reconsenting process, because that patient may actually want to continue in the study because the exposure has already occurred, and there's the ability to collect important additional safety as well as PK and dosing information. So in addition to the guideline that FDA published in 2017 about inclusion of pregnant individuals in clinical trials, a report was recently published by the National Academies of Science, Engineering, and Medicine. And this report was specifically to evaluate liability associated with enrollment of pregnant individuals in clinical trials. And the report's key findings are listed here, and somewhat surprising. And again, I would be happy to discuss this offline with those of you who might be interested. But one important finding is that there's a lack of evidence of liability actually related to enrollment of pregnant and lactating individuals in clinical trials. But there is potential liability after approval if pregnant individuals are exposed to those drugs, liability to the prescribers and potentially to drug manufacturers. And the report also concludes that liability could actually be prevented by including pregnant individuals in clinical trials. And I've given you the link to the report. It's quite a detailed report. And again, there are some limitations in these findings, but I think the report really highlights some perceptions that maybe need to be changed about inclusion of pregnant individuals in clinical trials. I also want to highlight ongoing work at the ICH level, that there is a guideline under development right now on inclusion of pregnant and breastfeeding individuals in clinical trials. And you can see here what the goals of that harmonized global guideline will include. So in the last few minutes, I want to discuss what FDA is doing to enhance diversity in clinical trials. So on this slide is a quote from our current FDA Commissioner, Dr. Rob Califf. And basically, Dr. Califf has made ensuring diversity in clinical trials an important issue for FDA to tackle because it is a fundamental public health issue. Now, in addition to his quote, FDA has been working to ensure the health of the whole public, including underrepresented populations, and that clinical trials should reflect the population that is most likely to use the drug. And that includes populations who have generally been underrepresented in clinical trials. So I don't have time in detail to go over all of FDA's efforts to ensure diversity in clinical trials, but suffice to say, you can see that FDA's been very busy over the last few years with guidances and workshops to help sponsors understand and incorporate diversity in clinical trials. I will also want to especially point out at the bottom the ADEPT 9, or the Advancing Development of Pediatric Therapeutics. Our 9th conference was specifically convened to discuss enhancing diversity in pediatric clinical trials. And this was just convened a couple of months ago. There was very robust discussion, and patient advocates as well as patients themselves discussing the importance of diversity in clinical trials. Just a quick word about how decentralized trials may enhance the ability to enroll a diverse population. So a decentralized clinical trial is just a trial where not all of the information is collected at the investigator's location, but some or all of the trial activities could be conducted at a remote location from the clinical trial sites. There are clear potential benefits, including patients not having to travel to those centers, take time off of work, find childcare, etc., that it can increase enrollment, particularly for rare sporadic diseases where patients may need to travel hundreds of miles to get to a site. And even when they are enrolled, it can improve continued participation and maintaining patients in clinical trials and avoiding dropouts, because some or all of those endpoints and measurements can be collected closer to the patient. Now, obviously, there are challenges related to that, including the need for training, as well as tracking strategies, because you're likely going to need more personnel in a decentralized trial. And then, of course, compliance issues that need to be worked out to ensure that the data collected are valid and reliable. So in summary, I hope to have conveyed to you that children, pregnant, and lactating individuals deserve to have access to medicines that have been developed with sufficient information to support their safe and effective use in these specific populations. That drug development should be designed early to include these specific populations and plans to collect data in these specific populations when it's expected that if the drug is approved, it will be used in these specific populations. Of course, that requires early planning. And just some, what I would say, catchphrases that I'd like to start hearing, and I'm bringing and presenting to you right now. That the health of the child begins with the health of the mother. And that children, pregnant, and lactating individuals are protected through research, not from research. And finally, that the U.S. population has become increasingly diverse, and clinical trials should reflect the population most likely to use the drug. And so there's a need to enhance the diversity of the clinical trials we are conducting for the specific benefit of all patients who could benefit from the use of an approved drug. Thank you very much for your attention. And before I forget, I have a challenge question for you. Specific populations that should be considered for enrollment in clinical trials during drug development include A, pediatric populations, B, pregnant and lactating populations, D, all of the above, E, none of the above. And I'll give you a minute to answer. And the answer, of course, is D, all of the above. Thank you again for giving me this opportunity to present to you today.
Well, thank you very much for these great presentations, Shabnam and Lynn. I think you've really very comprehensively covered the topic of safety evaluation early and late in clinical trials. And Lynn, thanks for addressing really some of the most important subpopulations, which are obviously addressed in regulations and requirements as well. So I'm going to move on to questions, and there are many of them, excellent questions from the audience. I'm going to start off with Shabnam. Shabnam, there's a question for you. What role do investigators play in ensuring patient safety during clinical trials? And what measures are in place to monitor and report adverse events? So this is about the role of investigators in safety.
That's a great question. Investigators play a key role, as you heard during the presentation, especially when it comes to reporting SAEs to the sponsor. Keeping in mind the exceptions that we discussed in the talk, investigators are involved in determining if the AE is serious, and you could refer to the list of questions to ask yourself about what constitutes the AE as serious. There were, serious is a regulatory definition, so it's clearly defined there. As investigators, you're required to provide a causality assessment for each of the SAEs reported to the sponsor, and that really helps the sponsor because the investigator's view is important when considering, when assessing the safety of the drug and determining whether to report the event eventually to FDA. It's the investigator who monitors the subject's response to the drug and is knowledgeable about the subject's clinical state. They know the medical history and the concomitant medications that the patient was on, and so they may be especially sensitive to distinctions between events that could be related to the drug versus those that might be due to the underlying disease process, for example. Over, Leonard. Thank you.
Thanks very much, Shabnam. I think obviously emphasizing very important points that probably nobody understands the adverse event as well as the investigator saw it. So obviously a very important source for information from us. So Lynn, I have a question for you. Interesting question which has come up here. If a trial protocol excludes pregnant women and a pregnancy occurs during a trial, what are the regulations or best practices for follow-up on the baby, including the length of follow-up? So what happens if somebody gets pregnant during a trial? Any thoughts there?
Thank you, Leonard, for that question, and thanks for the question being asked. So even when it's appropriate to exclude pregnant individuals from clinical trials, and I hope I've convinced you that we should move away from the automatic exclusion of pregnant individuals from clinical trials. But even when they are appropriately excluded, pregnancies happen. Not every contraceptive measure is 100% effective. And so I think the question is a very good one. Part of the importance is not to automatically remove the patient from the clinical trial, stop all follow-up, and lose that patient from potential important follow-up measures. So one of the things I think I mentioned earlier is the first thing that should happen is that the patient, if it's a blinded controlled trial, you have to break the blind on that patient because you need to know if that patient is exposed to the test treatment. After that, then what should occur is a question about what the risk benefit is to continuing that patient in the study, even if they are pregnant, and to reconsent them in that process. In other words, once you know if the patient's been treated, then they might be able to be continued in the study, at least for safety, even if we have to break the blind. But that requires the consent of the patient. So one of our best practices that we describe in the clinical ethics guidance that we have on pregnancy is to reconsent that patient and to continue to follow them on treatment if that's appropriate. And in addition, one thing that I think is very important is to collect PK data on that patient, because the PK data can help us understand better whether or not there needs to be dosing adjustments during pregnancy. Now, the second part of that question is what about the fetus and what about the infant after it's born? So again, depending on the situation, for example, with biological agents that are immune modulators, particularly if they're given in the third trimester, can pass and cross into the fetus through the placenta. And that can potentially have effects on the immune system of the newborn and infant. So depending on the drug that is under study, there might be a need to follow that infant, not just after birth, but for some period of time after birth, even and including up to a year or more. So we do have information about that in our pregnancy clinical ethics guidance related to pregnancy that I can direct you to. But it's a very good question. And there are a lot of things that can be done besides just saying that the pregnant individual has to be removed from the trial and not followed up. Thank you, Leonard.
Thanks very much, Lynn. I think an excellent explanation, emphasizing really the importance of getting the maximum amount of information about these situations that happen during the course of a clinical trial, both in pregnancy and obviously in exposed neonates. Shabnan, a question for you. Since an AE can include a worsening of an existing condition or may be new but appear gradually, when should the beginning of the AE be considered insofar as trial records are concerned? So I guess the question here is related to when do we regard the adverse event as beginning in relation to when the trial, the patient was put on the drug?
Okay, thanks, Leonard, for the question. So the adverse event definition, just recap that it's any untoward medical occurrence associated with the use of a drug in humans, whether or not considered drug-related. And adverse events that occur during the study drug treatment should be coded as treatment emergent to differentiate from events that may have occurred prior to study drug treatment. I think that's an important concept. Treatment emergent adverse events, they're coded in MedDRA with a flag, usually called a treatment emergent flag, and that would distinguish them from other AEs. For example, perhaps one that may have occurred prior to study treatment, perhaps a worsening of a condition even prior to receiving study treatment. If we're trying to distinguish AEs which may have been related to worsening of an existing condition, that did occur on drug, you may consider coding it in such a way that it reflects actual worsening of that event. So I hope that's clear. Over, Leonard.
Thanks very much. Okay, I have another one for you, Lynn. Would it be appropriate to recruit participants for studies to develop drugs for diseases that impact people from minority populations or children differently than drugs being developed for the general population? And the example that's given is this might be at hospitals, clinic, or disease support groups. So I guess a question targeted at drugs for diseases that impact people from minority populations. Any thoughts about that?
Yes, thanks, Leonard. An excellent question. One of the things that we have learned, particularly through that ADEPT 9 workshop that I mentioned in my talk, Enhancing Diversity in Pediatric Clinical Trials, I think it's right to state that, and as I mentioned in my talk, that, we need to study drugs in the populations that we believe are taking them. And so what we don't want to see necessarily is a drug development program for a disease that includes a substantial number of minority populations, that population is only studied in clinical trials that, for example, enroll a very non-diverse population. And I'll tell you why that's important. That's important because what we heard at this conference, this workshop that we convened, was that not only is it important to understand whether the drug behaves differently in a minority population compared to a non-minority population, but that when patients are recruited from distinct areas that have generally been underrepresented, that enrollment of those patients gives trust in those communities that this trial is worth something and this drug, if it's approved, is actually acceptable to take. We heard from many stakeholders, including patients, minority and underrepresented populations, who said that, why would we take a drug that was approved by FDA? Because we don't trust the government, and nobody in our neighborhood even entered the clinical trial. But if there's enrollment of patients from those underserved areas and those patients go back and talk about their experience, not only does it help with recruitment in those areas, but it helps after, as I said, the drug is potentially approved and available, that it gives confidence and trust that those drugs are actually safe and effective, as FDA has determined that. So yes, I agree that it's important to enroll a diverse population, including those that might be historically underserved. And yes, those sites can include regional clinics. They can include engagement with disease support groups. And again, that idea of the decentralized trial is also one that can help with enrolling populations that have been historically underrepresented. Thank you.
Thanks, Lynn. I think you make an excellent point that inclusion improves trust in communities that are not generally participating, a really important consideration. Shabnam, I have a question for you. The question's simple. I think the answer has nuances. What kind of study will have death as an endpoint? Do you want to respond to that one?
Yeah, sure, Leonard. Thanks for the question. What kind of study would have death as an endpoint? Death is an endpoint for several types of clinical trials, and especially for diseases that are considered serious and life-threatening, it can be very useful. An example of that would be assessment of an all-cause or a 14-day mortality, which can often be an efficacy endpoint. Again, in trials that are looking at conditions that are serious or life-threatening. And in the anti-infective world, one example could be phase 3 trial for example, for a healthcare-acquired or healthcare-associated or ventilator-acquired bacterial pneumonia, in which we often have efficacy endpoints looking at mortality at a certain time point to assess the non-inferiority of one antibacterial drug versus another standard of care antibacterial drug. And then in the talk, as you recall, the example was for an antihypertensive drug that was looking at endpoints related to mortality and acute myocardial infarction and stroke and that sort of thing.
Yeah, thanks very much. Sorry, Shabnam.
Yeah, that's it. Thank you.
Yeah, I just had some thoughts as well. Outside of the realm of infectious disease, obviously cancer trials are very often have mortality as an endpoint or time to death, because many of those patients do experience death. The other thing that probably is just that came to my mind was sometimes difficult to tell whether death is the endpoint as a result of a failure of the drug or whether it may be some other cause. And, I think sometimes evaluating death may be tricky, particularly if patients have serious underlying comorbidities. So I think it's a very insightful question. Let's see. I'd like to move on to another one for Lynn. This one's about dosage extrapolation, Lynn. The question says, we can't extrapolate dosage in neonatal populations because of the very different physiology related to gestational age, right? Any answers to that?
Yes, I do, Leonard. Thanks for that question. Again, another very insightful question. So it is true that, and again, in my discussion of extrapolation, it relies on how similar the population is with the disease, a particular pediatric one, compared to, for example, an older pediatric population or an adult population. And we certainly understand that neonates have a very rapid change in their physiology, their metabolism, their physical growth over a very short period of time. So it is hard to imagine that neonates could be similar to an adult population, for example. So we'd have to absolutely do separate neonatal trials. And the answer to that is that, you know, it may be true that trying to compare a neonate to an adult would be difficult, but comparing a neonate to, for example, a 6-month-old or a 3-month-old, and if we have dosing information and efficacy established in those groups, it may be very reasonable not to have to conduct, you know, a fully powered, randomized, placebo-controlled trial in the neonatal population. But leveraging what we know about that 2-month-old or 6-month-old population, that we would still need to collect probably safety information, and yes, to your point, dosing information, because we recognize that those metabolic enzymes develop rapidly during the neonatal period. So yes, we would likely need to have dosing information, and we would likely need to have safety information, but probably not necessarily in all cases separate neonatal randomized controlled trials. And the only other point I want to make too is that if, for example, this drug had been tested in neonates for a different condition, and now the same drug is being looked at for yet another indication in neonates, then having that information from another previous development program in neonates can be very helpful, and that information could potentially be leveraged. Thanks.
Thank you very much, Lynn. As a non-pediatrician, I can only imagine how difficult it is to study neonates for the activity of new drugs. Shabnam, I have another question for you. How is the term severe used in the classification of AEs?
Yeah, thank you for the question. I'm glad the question was asked because this does come up a lot. And so, you know, I had mentioned in the talk that severe does not equate with serious, as that is a regulatory definition. But severity can be an indication of intensity of a specific event. So that's the key word, is the intensity when we're talking about how severe something is. Severity can be assessed by those toxicity grading scales that I had mentioned. And you can, you know, reference some of those examples on the slide just to take a look at what may constitute mild, moderate, and severe for different patient populations. So in general, you know, severe AEs would, as you would think, would be those that would cause significant pain, discomfort, for example, or may prevent daily activity, as an example, depending again on the study population. What may be severe for one patient population, like a healthy volunteer, may not be the same for you know, oncology patients, for example. And then in terms of laboratory abnormalities, a severe AE would probably be one, you know, obviously that's worse than mild and moderate in terms of the actual, you know, normal reference ranges, and it might be one that might require medical intervention. So, so I think hopefully that is clear. Thanks.
Thanks very much. Again, let's just alternate. Lynn, a question for you about pediatric extrapolation. Thoughtful question here. For pediatric extrapolation, can you use modeling and simulation before starting your trial for a better informed program?
Yeah, thanks, Leonard. That is a really good question and a very important one. So as I mentioned, you know, extrapolation, this idea of extrapolation is to leverage what we know about, you know, a reference population, you know, an adult population or older pediatric population, and apply it to that population that you're interested in studying, which is usually a younger target pediatric population. And the whole notion of modeling and simulation, that is to take data that we have and to understand those relationships within the data using computational methods. That idea of modeling and simulation is a critically important component of the analysis of data. Not just, you know, people say, well, of course we do modeling and simulation to derive a dose, you know, if we want to model a dose in children that could match an adult dose that was effective. Yeah, that's clearly something that we have and that we do in even overall drug development. But one of the things where I want to point out another important place where modeling and simulation can be useful is in evaluating the data that we have that even supports similarity between an adult and pediatric population. So you may have data from different published studies. You may have data from case studies. You may have data from registry studies, and you may have animal data, for example, that can help be used to support a similarity of the disease between a pediatric and adult population. Well, these types of data and using modeling are tailor-made to understand the relationships. So I think that this is a great way to use modeling and simulation. In addition to the uses, once you have established an extrapolation concept and want to figure out dosing, et cetera, as part of a plan. I'm sorry, I'm losing my voice, but thank you.
Thanks very much, Lynn. Are you up to another question? We'll see how your voice holds up. But this one,
I just took a drink, so I think I'm okay.
Okay, I'm glad you recovered. So here we go. I think this one it's a great question because it opens up a couple of important points for discussion. So what are some ways to address challenges in conducting clinical trials in pediatric patients? For example, how can you obtain sufficient information when the pediatric population available for the study is very small? Any thoughts about that?
I do. And in fact, we convened a workshop 2 years ago on innovative trial designs that specifically could be considered as part of a pediatric drug development program. And by the way, the reason I think that this topic is so relevant, even if you're not interested in the pediatric drug development per se, is that the same principles that apply to developing drugs for small populations also applies to rare diseases. So many pediatric diseases are considered relatively rare compared to the adult population, just like rare diseases overall. So the same principles and the same strategies and the same innovative trial designs that we use in pediatric drug development can be applied in many cases to rare disease drug development. So the idea of Bayesian approaches, the idea of, externally controlled studies, the idea of N=1 trials, all of these potential strategies, and of course the use of modeling and simulation to help decrease the number of patients that you might need. All of these approaches are discussed in the ICH E11A guideline that I described to you and is available now on the website for ICH. So it's really important to consider, and we have many different approaches, and that guideline, I think, really provides excellent examples and references for you. And again, I would encourage you to read that because if you're interested in rare disease development, some of these same study methodologies and innovations also apply there. Thank you.
Thanks for a great answer. I guess emphasizing that one really needs to be as creative as one can at getting the information that's needed. We're running close to the end of the session. Shabnam, I have one other for you. I think this is regard to safety information. What type of information would go into the investigator's brochure?
Okay, thank you, Leonard, for that question. So the investigator brochure or the IB should have in it all the information that's important for the investigator who's administering the drug to human subjects to really know and understand. And it's required that the information include information on drug substance and formulation and the pharmacological and toxicological effects of the drugs and then again in humans if known. And then obviously information relating to safety and effectiveness in humans obtained from prior clinical studies, and that's going to be key. Also, possible risks and side effects that are anticipated on the basis of prior experience with the drug under investigation, and then also experience that are you can have from related drugs in the same class, for example. And then lastly, it should include precautions or special monitoring to be done as part of the investigational use of the drug. I think there's another talk on investigational brochures tomorrow as well. Thank you, Leonard.
Great. Yes, I think some of that will be covered. Just one more for you, Shabnam. Interesting question. In the hypothetical case, does the investigator report death or anaphylaxis or both? I presume if the patient has an anaphylaxis and dies, how does that get reported? Any thoughts about that, Shabnam?
Yeah, thank you for the question. So, the investigator would report to the sponsor this AE as a death, and the cause of death, should be clearly communicated as due to anaphylaxis. In this case, death by itself would not be very informative without letting the sponsor know what they think is the cause of death. And in addition, a causality assessment by the investigator should be provided to the sponsor. As much information about the event as you could, note would be helpful to the sponsor, who would then ultimately determine when and whether to report to FDA in this hypothetical example.
Great. Thanks for the answer there. So I think that pretty much brings us to the end of this session. Again, a vote of thanks to both of you for excellent talks and Q&A session addressing safety and addressing specific populations. I think it brings to the close our content for today, which is really focused on clinical trials and what happens in clinical trials. Tomorrow we'll reconvene, and I think the topics that we'll begin with are non-clinical and early clinical drug development, which we haven't discussed yet. They include chemistry and manufacturing controls, pharmacology and toxicology, and clinical pharmacology, all very important topics that obviously we base our design of clinical trials on. Following that, we'll have a couple of lectures, one on ethical considerations, which we haven't touched on yet. I'll be giving a short lecture on innovative trial designs and approaches, and then we'll finish off tomorrow's sessions with two talks on very innovative treatment modalities that are just coming to the fore and offer a lot of promise for diseases that we really couldn't deal with before. So an exciting lineup for tomorrow, and I'm very much hoping that everyone will join us again. And at this point, I'm going to hand over to Brenda, who I believe may have some closing housekeeping announcements. Thank you.
Thank you, Dr. Sachs. Well, we have come to the end of today's sessions, but fortunately, like Dr. Sachs says, we have 2 more days to go. Just as a reminder, to simplify things, each day SBIA will send one email to the attendees of that individual day, and that email will have information on both the CE claim codes for physicians, pharmacists, and nurses, and also the survey link by which you can download a certificate of attendance. We will send that out within 24 business hours, so please look forward to that tomorrow. Both final activity evaluations, CE and event survey, must be completed within 2 weeks after this activity, no exceptions. So for this event, the deadline date is December 26th. I would like to thank Dr. Smith and Sachs for the spectacular job done as hosts and moderators, the faculty for all their presentations and insightful Q&A panels, and thank you to the audience for being with us today, for your commitment, attention, engagement with your very, very deep questions. Please join us again tomorrow. Invite your colleagues to join us as well, and we look forward to seeing you again at 11 a.m. tomorrow. Until then, see you later.